To my sadness, I've tried everything I could to get Bob not to retire, and he was very adamant that he has a plan. So I have to honor that. What I wanted to do first was realizing something about someone with Bob's background and time of being a scientist and training people, is that the professor is the only person who knows everyone that goes through the lab, right? And the lab actually has generations. So one of the places I wanted to start with first was to have Bob and Liz stand up. I assume everyone knows who they are, but I would like to have them just stand up and wave so everyone knows who we're talking about. So they're not here alone. Their family members are here. I'd like those folks to stand up. So this way, if you're sitting with somebody, you know the connections. And now all of the lab alumni, past, present, and future, please stand. Okay? So these are the folks that actually helped Bob along the way. And then I'm guessing everyone else is colleagues, friends, and other. So I won't have all you stand up at this point. Unless you want to. Are there any enemies? Okay. I want to just very quickly go through a few facets of Bob, right? His science. We all know him from PARC. We all know him from thinking about how light gets harvested. And actually, the painting of the light waterfall is probably one of my favorite pieces of artwork that actually really explains Bob's science in one visual image. And, go back through his more than 400 papers, not quite 500, but pretty close, right? Thinking about how that water mill works, right? And how you can take sunlight and turn it into something that's so powerful, that's the basis of everything on the planet. It is really amazing. Bob is also a fantastic teacher, having watched him teach Biochemistry 1 and his writing a textbook on photosynthesis, right? I always tell people, if you want to know something about photosynthesis, don't ask me, go straight to Bob. He's the source. And I know that his students, both in the lab and outside of the lab in the lectures have just really appreciated everything he's done. And in fact, this is Bob's favorite lecture room. So this is one of the reasons we picked this spot for the symposium. Now, as a mentor, right, all of you that stood up at one point know what he's like. I couldn't jam all the pictures in here, but, a long story of many different individuals have gone through his group. And I'm waiting for his last student to show up because he's supposed to be emceeing this part of the morning. So we have to reprimand Ben a little bit this morning. He'll get here. And I just want to sort of frame a little bit of what's next. We all know he's moving to Arizona. But it was last fall I gave Bob a personal going away gift because his connection to photosynthesis is so deep. I don't know if many of you know this. There's actually a game for photosynthesis. So I gave him this as my personal going away present, right? Welcome to the world of photosynthesis, the Green Strategy board game. Plant and shape the ever-changing forest as you cultivate your seeds and your strategy. The game actually has a sun that is here, right? And your different teams planting plants at different stages, competing for that sunlight. I think Don Ort's talk will tie into this quite nicely. And you have to take those trees through the life cycle from seedling to bloom to rebirth and earn points while collecting the energy and revolving the sun's rays. So hopefully while you're in Arizona, soak up the sun, play the game, have fun with that. Really? I actually haven't played it. I haven't played it yet, so I was hoping it'd be good. And hopefully your scientific knowledge doesn't give you a cheating advantage on things. It's the opposite. Okay, it blinded him to how things actually work. So really the idea behind the symposium is to celebrate Bob, and we're trying to be a little informal. I want to give, everyone should have gotten a piece of paper with the overview. Basically, we're going to start with a little bit of history of Bob by Gavinji. We'll tackle Bob's first graduate student, Barry Bruce. A little bit about photosynthesis from Don. We'll take a break. And then traveling with Bob through Earth's early photosynthetic history by Carl. Thinking a little bit about green history with Gary, who'll be joining us by Zoom. So I'm hoping that all works when we do the switchover. And then the last graduate student and lab members have decided to do a tag team talk, I believe. And then we'll have some lunch instructions. You'll notice the tent outside. That is where we'll be having lunch and margaritas. It'll be barbecue and margaritas. We thought that was very Bob. That is also why you have those tickets in the back of your name tag. And then we're going to have Mike Gross talk a little bit about collaborating with Bob, and then flash talks by lab alumni. So I think I have most people's slides up here. Also in here, I'll open it up sort of in the style of a Quaker meeting house, that if somebody feels moved to say something about Bob, stand up and just say what you want to say. And then we're going to leave the last word for Bob and Liz to say what they would like to say about all of us. So we hope you guys enjoy the day. And with that, I'll get ready to have Govinci come up. And Ben Wolf will be moderating the first sessions.
Okay, so I'm very delighted to be here. Very, very delighted to be here, very happy. So Bob did his PhD under Ken Sauer, and it took me several days to get a letter from Ken. And it is also signed by Ken. So I will not read it. So I have two copies, one that you can read, the other I made cursive so that you can enjoy it. So I will give you, before my talk, this letter from your professor. All right, so now with that, let us look what I have. I have two names now since last year. It's called Govinci. And since I'm called Gov by Bob, so I put Gov Govinci. That is the explanation for this slide. And you know where he's going, so there is a place, Arizona, where he's going. What is this place? You know Bob very well. Right. So that's the beginning. Now let us look at this guy, how he was. That's laughing. You were Larissa, right? You have seen him, right? All right, so these are the pictures of Bob. So look at Bob when he was a baby, then look at the handsome young boy. We need to celebrate August 25, I hope is the correct date, his birthday, which is coming up this year. Every year it comes, and it is into Independence Day, with July 4th and August 15th, the Independence Day of India. Now look who are these people. So this is just for fun. Now, look at the handsome boy. And look at the brothers and sisters. All right, Bill and BJ. What a glow. What a glow. Look at him. All right. This is the best picture. This is the best. 1971. And there are a lot of stories about this. I will not have time. I should be given 2 hours to talk about Bob. Unfortunately, 20 minutes will perhaps be certainly not enough. But let us look at him. It's a wonderful day. And Liz and Bob, with lovely couple, the Golden Gate Park. They can now visit this park because it's closer to Arizona. All right. Now, here is a British Mafia with him. So who are they? Les Dutton, whose name doesn't appear on my slide for some reason. I don't know why. Is there a reason? No. Just missed it, not enough space. Richard Cogdell, Colin Raitt, our dearest friend of all, who has left us. So remember the British Mafia. That's wonderful. The experiment. What a hairdo. And a look in his eyes. He has been a great experimentalist, as we all know, Stoll, as well as a theorist. In my personal, private opinion, his experimental work is above that of Robert Emerson. Believe me. And his theoretical work is above that of Eugene Rabinovich, two giants of photosynthesis of my time. They were my professors and also of Rajni's professors. So I really, really appreciate what Bob has done for us. Now, look at this. Now, who are these people? There is Bob, and there is Anna Moore, and there is Liz. Look how cool. So look at this. What about people they will meet at ASU? One of them. There will be many. And this retiree from whatever say, Busl, WSTL, may want to set them right. She would say, okay, who is she? You settle the argument, that's Liz. I am staying out, as she once did in 2013 when Anna Moore and Bob were discussing a scientific. Look at his eyes. Boy, you have to agree with Bob no matter what. All right, now let's read this. I know that your publications, mean Bob's publications, are like the former Nizam of Hyderabad's children. He had 500 children, that guy, Nizam. He had 430 papers. So instead of having 500 children, he had 500 papers, close to 500 papers. That's the similarity with the Nizam of Hyderabad. So sometimes you need to be ruthless and choose just a few to talk about, which I will do. Now, I must say one more thing. People don't know this gentleman. His name is Daniel Arnon, discoverer of photophosphorylation. So he had to be once, literally, dragged out from this place, podium, not in this room, another place. So please forgive me, and I want to be a little bit like Dan Arnon today. I maybe exceed my 20 minutes given to me. All right, so this slide was prepared when he got an award at Urbana-Champaign called the Rabez Foundation Award, but I couldn't be there. All right, now a few papers, and with few people that we know. Jerry Babcock, with Jerry Babcock, and this is a paper, Blankenship, Babcock, Joe Warden, and Ken Sauer. And I won't talk about the paper, just to show you the picture and the title. He was a fellow student. Again, another person to remember, Jerry Babcock. Both of them came for my retirement in 1999, I assume. And I remember both of their talks. All right. Here's another guy. I think Don Ort perhaps knows this fellow too, who's sitting. So Bob worked as a postdoc. Magnetic field effects on radical pair intermediates in bacterial photosynthesis, my God. And so he's a wonderful guy too. So Bob had the privilege of working with wonderful people, Ken Sauer and Bill Parson. Here's another guy, Howard Guest, a good friend of all of us, and Clint Fuller. So they discovered a unique reaction center from Heliobacterium chlorum. Mine. So late Clint Fuller is over there, and our friend Howard Guest is over there. Here's, really they are here, looking at Bob, say, hey, great, you are retiring. All right, so this is the fun slide that I really enjoy and use in all my lectures all over the world, especially in India and China. So this antenna, energy transfer, electrons, the real stuff that converts light energy to chemical energy. So that's my favorite slide I made. And I won't talk about this, it's very too complex. He's a world authority in evolution. Extreme diversity event in a system has suggested to him multiple independent evolutionary origins. He also looks at reaction centers. See, right for his earlier concepts of evolution, don't try to read it now. Just read it in the paper if you are interested. Oh, let us look at this. We mentioned his wonderful molecular evolution analysis, a complete genome from 11 known phyla of photosynthetic prokaryotes, blah, 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 published with Raymond et al. So enjoy this picture and read the paper later, if you wish. He came from Arizona State University. He came here in 2006, I'm told. And so this was just before he left. And look at all the young people. I think some of them are here. I can recognize some of them, their faces. And I don't know whether Martin is here or not. Where is he? Where are you? Good to see you. Are you still in Norway? Oh, good. I will come and visit you. All right, so there are these wonderful people. Some of them are here. I thought I'd show you his picture. All right. I was there when Bob was recognized here at this university. And he became a Lucille Marquis Distinguished Professor of Arts and Sciences in 2006. And to be sure, you know I was there. All right. Now here, and here are his, are you all here? Some of you are here? Yeah, good. So I'm glad I found your pictures and this group. So he's trained many students, just a small group, 2007, and I call his gang, and the gang is here. All right. Nancy, where are you? Are you here? Nancy? Oh, God. She should be fined. She was there. Yeah, I know. Oh, there. See. So we caught you. Not allowed to go out of the room, you know. So in 2007, together with Nancy Kiang, who's here, and NASA, we went searching for life and photosynthesis in outer space. And here he's still looking, may join them next year, who knows. So Nancy and Bob and I, we wrote a paper and what would be the color of plants in outer space. And Nancy decided to write a beautiful Scientific American article, which I hope she remembers that I read it before you published it, right? So it was wonderful. The color of plants could be red, blue, and even black. The Nature wrote an article on our paper and says, could be orange. We don't know what it is until we go to Mars and see it. All right. So now, 2009, with Lars Olaf. Now I want to be sure you recognize me. There's a tree behind me doing photosynthesis, and Lars Olaf inside. So we wrote a paper, Why Chlorophyll A? And we had a very nice paper telling, so read it, and you will tell you what we thought, why chlorophyll A was invented to do what it is doing. All right. I don't want, it's published in Science. It's a very important paper. And some of you authors may be here. Bob is certainly here. And phycobilisomes supply excitations to both phosis systems in a mega complex. This is the paper. It's a beautiful paper. Enjoy it. And this person is here, I'm told. Yes? Are you here? There. So that's a discovery just 3 years before his retirement. My God. And wait for 3 years later. There will be another discovery at Arizona State University. Watch for it. I hope I am there to talk about your next discovery. So I won't discuss the discovery. Here in this paper, they discovered in FMO, Fenner-Mathews-Olsen protein, full form, a new type of energy transfer, regulation, providing for a novel energy quenching mechanism involving cysteine microchlorophyll photochemistry. It's a wonderful paper. Read it, and you will enjoy it. Oh, sorry. Now, we have seen this big book. Bob wrote a really great, thorough, and authoritative book, an outstanding service to education around the world. Even Ken Sauer, his former advisor, thinks so. That is something important. If you're a professor, think so. And also, and I tried to tell Don Art, my friend here, why not use my book? He said, no. Bob's book is much better. So you can see, I'm having great fun. And I have read this book 3 times. All right. Now, I thank everyone who helped me prepare this talk. I won't give their name. More importantly, Joe for inviting me, and Amanda for making arrangements. Special thanks. But there is another slide. Where is it? Who is in charge? Oh, so I just keep going? Okay, there are few more slides. So I wrote to Vitalia Chandra, and I wrote to Ken Sauer, and I telephone, and they send hundreds of pictures. Vital sent hundreds of pictures on Bob. Now let's look at just 2 of them, and then I will stop. Okay, he has different looks for different friends. This I didn't know. Look at this one. Now this is Rob Burnap. Look at him, very serious. You are right. And this is, I think, Vitalia Chandra when he had a beard. Is that true? Okay. And look at Bob's eyes. Really? Well, now look at the third guy, Chuck Dismuth. our good friend in photosynthesis, very, very energetic. And he says, but see what Bob says? I don't believe you. I think that's, oh, no, one more. That's the last, I think. So when Bob lectures, you must not sleep. Never sleep. You can never sleep, even if you want to sleep. Why? If you did, he will send you out to the exit door. Bye-bye. No more coming in the class, failing grade. So I think that is my last slide, I think, because nothing else is coming. Thank you very much.
Okay, sorry. By Beverly Pearson and Richard Castenholz, Chloroflexus aurantiacus. And my project was to look at the primary photochemistry of this. And I was a relatively new student, but I knew a little bit of spectroscopy, having worked in a physics lab at UC Santa Cruz. And so we basically set out to do this. And at the end of the day, we essentially discovered a very interesting thing. Chloroflexus is a purple bacteria in green clothes. And this was completely surprising to all of the people in the prokaryotic world of photosynthesis. And it essentially argued that this may occupy a really fundamental place in the evolution of photosynthesis because it essentially has this electron transport chain very similar to the purple bacteria, but it has a light harvesting antenna that involves chlorosomes, something that Bob really was a pioneer in working on. I left that group. This is a picture that sort of shows an interesting transition. If we go back, I just want to point out this paper was communicated by Bill Arnold, who is at Oak Ridge National Lab of Emerson and Arnold's notoriety. And it was very interesting. I was a young scientist, and I was submitting this to publication, and Bill Arnold insisted that he speak with me. And so I called Bill Arnold. I sat in Clint's office, and he interrogated me for about a half an hour about all of the work, because he was going to communicate this, and he wanted to have a personal knowledge as to who was doing the work and what was the quality of the work. But I got to know this person vaguely, I guess. But then when I left, as I said, it was in the chemistry department, and this was a stupendous program of people. None of them are there now except, I don't think any of them. And I actually called all of them to try to get photos. They all gave me nice words to say about Bob, but none of them. Lila said I should go and talk to Alan Wagner because he was a photographer, and he said go talk to Dave Dooley because he's an administrator, and then go talk to Alan Croft, but Alan Croft has passed away, and Ruth Stark. But this entire group of people were all assistant professors at Amherst College, and they are all stupendous scientists. But I worked with Clint Fuller, and this is Bill Arnold here, and it turned out that this was a key thing because when I actually went on to get a job, it was my connection with Clint Fuller and Bill Arnold that actually landed me this position. Out of hundreds of applicants, the fact that I had this knowledge of a person, because I went to UT Knoxville, which is right next to Oak Ridge National Lab. And so there's a certain serendipity there that sort of connected me there because the department head, Wesley Wicks, Clint Fuller, and Bill Arnold all worked together at the ORNL. I left at Bob's lab and I went to Berkeley. And when I got to Berkeley, there was this project going on that was between Alex Glazer and Dick Malkin, who I worked with. And this is something I didn't really think about when I was doing it, but they were trying to elucidate the subunit stoichiometry. This is really pre-crystal structure. What did the photosystem look like? What was the structure of photosystem 1? And they did it by isotopic labeling where they could really get a very clear ratio. And they came up with this model that the Synechococcus PS1 complex was a tetrameric structure. And this was a bit bamboozling to a lot of the people. That this thing was a tetramer because at this point they felt that PS2 was a somewhat dimeric structure. But this was the project, and then I said, well, I'm going to reinvestigate this. So I started working with a plant that we could do in vivo labeling with. This is a plant called duckweed or limna. And essentially I worked with Dick Malkin, and essentially we did the same thing. We did it maybe better. Maybe we did it with a little bit more genomic data. But effectively we came up with a true dimeric model. This was profound in that it essentially changed our sort of primitive architecture drawings, and it basically allowed us to conclude that the PS1 structure has a molecular mass that would be consistent with this trimeric structure and not the tetrameric structure. It also gave rise to the fact that it forced us to recognize the nature of FX, which at that time was unknown, whether it was two iron sulfurs or 1 4-iron sulfur. So this was really the last time I worried about the structure of PS1. I left the field I went off and worked in chloroplast protein import, and then started working in an area that was also serendipity with a person, Elias Greenbaum at Oak Ridge National Lab, who was interested in making hydrogen, and so my lab decided that probably the best system to work on hydrogen would be Photosystem 1. So in about 2001, 2002, I basically started making Photosystem 1 again after about 12, 15 years. And we had a whole rich opportunity in doing what I call applied photosynthesis. But at one point I said, I want to work on an organism that's going to be more thermal stable. We were working with Thermosynechococcus elongatus that comes from the Beppu hot springs in Japan. And I looked around and around, and I could only find one other report of a thermophilic cyanobacteria that could be grown to high density. And it's this relatively esoteric organism called Crocosidiopsis strain TS821. And TS stands for thermotolerant strain. And this was not a strain that was in any of the Pasteur culture collections. It's not in any culture collection today. As near as I can tell, it's only in my laboratory. And to get this, I had to actually go to Thailand. So I flew to Thailand. I went to Chiang Mai. And again, another somewhat serendipitous is that this project was actually sort of, again, motivated by work by Dick Kostenholz, which was the sort of the paternal father of the Chloroflexus that I'd done my graduate work on at this point 20 years earlier. And so this is just showing some of the organisms that are found in these hot springs up here near Chiang Mai. These are very unusual hot springs. Their flow rates are completely linked to the weather. They're called monsoon hot springs. They're dry in the dry seasons, and they really are flowing very quickly. So I went to Thailand. This is the Dr. Pir Panopisilal that is the collector of this, and it's quite remarkable. This woman has over 600 strains of thermophilic cyanobacteria in her laboratory that virtually nobody in the world has ever studied. But because I had a PhD student from Thailand, I was able to go there. She didn't speak English. She didn't use email. But fortunately, I could get this, and I brought it back to Tennessee because it was a thermophilic cyanobacteria, and I was very interested. Now, it is put into a group of cyanobacteria that are called the Plurocapsales. And this is a group that was characterized first by Waterbury and Stanier in the mid-70s. And it had a lot of interesting features, and the most interesting one is it undergoes binary fission in 3 planes. And so it can create this cubical structure, and it is surrounded by this very heavy, dense, carbohydrate-rich material that gives it this name capsule. And this is a really rich system, and it has a very different ultrastructure. So here's the inner membrane, the outer membrane, the peptidoglycan, and then it has this layer that's called this F1 layer. We're still actually studying the F1 layer, but the F1 layer is probably the source of its extremophilic tolerance. And so we got this organism. It was called Crocosidiopsis, largely based on this anatomical or morphological trait. This is what it looks like. This basically will make these 8-cell structures. They seem to stick together because there's a lot of mucilaginous material. But it turns out that people have looked at the Crococales and have shown that these are very present. They were quite durable for a cyanobacteria in the fossil record and have been observed all the way back to about 2.6 billion years ago. And so there was this thought that the Crococales, Crocosidiopsis, are really, really primitive cyanobacteria. There's a recent paper that has shown that they are basically cosmopolitan. They're found all over the globe. They're found in very cold settings, very hot settings. A lot of this was thought to precede any of the plate tectonic stuff. So we were very interested that we had this old cyanobacteria. When we then isolated the 16S RNA and tried to figure out exactly what it was, we discovered sort of an interesting thing. Here are all the Plurocapsales, and this is Myxosarcina, Dermocapsa, Plurocapsa, all of these guys. There is one Crocosidiopsis, but our Crocosidiopsis sits up here, not with any of the rest of them, but in this unique clade that is close to the heterocyst-forming cyanobacteria. So we have now formally renamed this. We call this the heterocyst and close relatives, HCR. You'll see more about this. And it's not related to any of the other members of the Plurocapsales, and it's very far away from TE or Prochlorococcus or things like that. So at this point, we wanted this for photosystem 1, of course, because that was the system that we were putting in our solar cells. And at this point, all of the photosystem 1s known were trimeric structures, just like they were in T. elongatus or 6803. And so we were going to isolate this thing, and we had a real difficult time. Okay? Essentially, it did not behave like the normal PS1, and so we started to look at its structure using Blue Native Page. And here is T. elongatus. This is essentially a titration using different amounts of dodecyl maltoside with a blue native page. And in every condition, we had this much larger complex, 1.6 megadaltons. This is the trimer. We always had something that looked very much like a dimer, and then finally something that looked like a monomer. We had nothing that looked like a trimeric form of PS1. We had to fight, we had to do a lot of work, but eventually we published this in the Plant Cell, and we convinced the community that there is, in fact, a trimeric form of PS1 that's not an artifact from detergent solubilization, because we could actually see it in non-solubilized thylakoid membranes. Turns out about the same time, there was a group in Japan working on a heterocyst-forming cyanobacteria nostoc, and they observed the same structure. So now Crocosidiopsis TS821 has a tetramer. These guys working with Anabina had it. They actually have a very interesting thing that they found in many of their cases. It's actually attached to probably a phycobiolysome rod. And Bob is a big advocate of this understanding of how phycobilins act as accessory pigments and how they move with their linker proteins, the CPC G1 and G2. And here's a cross-section of what may be a rod structure attached to this. And we're actually exploring this in our own lab because we think we also have these same structures that we can observe. It's probably not quite like that, but somehow in this tetramer we're getting a phycobilin. Now, when we looked at the genomic structure of these organisms, we went back and we had quite a few sequences available. Here is our organism. Here is the nostoc or Anabina. These are all tetrameric forms. This is a trimeric, so here's a different Crocosidiopsis thermalis. Here's 6803, here's T. elongatus, Syncococcus, Prochlorococcus. They have a very different genomic structure of their photosystem 1 genes. These all have a PSA F, J, L, and then very interestingly, they have this GMK gene that's in opposite orientation. The trimerics all come with a different, some random piece of DNA here, but then they have PSAL followed by I, or sometimes I is in front of it. So these two different structures in the genome seem to be closely correlated to this, and we'll come back and look at this in a little bit more depth, the FJL GMK structure in a moment. So another form of serendipity is I was invited to go and teach science for the Dalai Lama in Dhammasala. This is a very unique opportunity. I spent about 3 weeks teaching the secret, this is if you really could look in here, I taught him the central dogma of molecular biology theatrically, and these are all actors. Each of these were an amino acid, a tRNA, things like that. I'm sitting over here. But en route to going to Dhammasala, I basically stopped in Paris and spent a few days. And I went to the Pasteur Culture Collection. And I essentially went there, foisted myself on them, and persuaded Rosie Ripka and Muriel Gujar, the current curator, that we had a very interesting story and we wanted them to work with us because we simply didn't have enough capability to have this many cyanobacteria at our disposal. And so with their help and their ability to grow and let us have access to their 1,200 different cyanobacteria, my graduate student, Meng Li, started to characterize 62 different cyanobacteria. We did it first by blue native page, so these are all the tetrameric species up here. This is just a small snapshot. We had to do 62 blue native pages followed by 62 silver staining gels to confirm all of this, but it was very clear that there's a population that is tetrameric, and there's a very clear that there's a population that's trimeric. This can be easily reported by a good blue native page system. In association with Genesis, a bioinformatic group that's at the CNRS, we then went back and built a new family tree of all the genomes the Pasteur Institute had access to. At this point, I think it was just over 200 fully sequenced genomes. And we basically color-coded this in two ways. This is the gene structure I showed you here, the FJL structure. This is the LI, and then sometimes there's this orphan L that's shown here in light blue. So in these colors, this is the genome structure, and then everything that's shown here with either a triangle, a half circle, or a full circle have been tested for their oligomeric state. And what you can see is that this group right here, which we now call the heterocyst and their close relatives, all have this altered genome structure, and in almost every case that we study, we didn't study them in every case. Ones that are lacking here, we just didn't actually investigate, but all of these have either a tetrameric or a dimeric. And the tetrameric and dimeric is a bit of an operational distinction. We believe it's a tetramer, but it's a relatively labile system that's truly a dimer of dimers. So we felt that we now have really been able to identify in this group here, which is blown up here, a subclase. Now there's some interesting things up here which look to have it randomly. We'll look at these later. This is the pseudoanabina and things like that. But generally, all of the other trimeric guys really stand out quite closely. So we wanted to sort of try to decide what was the purpose of this. And so since the heterocyst organisms are nitrogen fixing, and this is a diurnal nitrogen fixing organism, we did all these physiological studies. But the only thing that led us to any observational correlation was light intensity. So if we grow them at low light, 40 microeinsteins, and relatively high density, the cultures make essentially very little tetramer. If we raise them and grow them in less dense cultures, they make a large amount of the tetramer. They also upregulate their carotenoid content a great deal. You can see that this is a low light culture, mostly monomeric and dimeric, maybe a little trimer right there, you can see. But in high light, it's really been pushed up into the tetramer. And you can see this both in the blue native page and then staining it, you can see here. We did that for 2 other organisms, Glyocapsa and Fischerella, and the same observation observed. So the tetrameric form of the PS1 accumulates when grown in high light and high light intensity, as do the increase in the carotenoids and the thylakoids. We then did so this is a little bit of a correlation. This is the tetramer, this is the trimer going down. Here is a different spectra of the thylakoids grown in low light versus high light. You can see some increase in carotenoids. If you take the different spectrum, you can get a very clear spectrum showing the increase in the accumulation of the carotenoids. But then we did an interesting thing. We took the tetramer and we developed a biochemical way to crack that egg open. We could basically pop it in half. And when you do that, when you go from the tetramer, this is cracked open and run on a second gradient, there's a whole lot of free chlorophyll that now runs at the top. The free chlorophyll has somehow been released and may, in our model at least, sit within the donut hole. The donut hole is essentially a carotenoid-rich compartment that when you break this in half, that is possibly lost. So we then went and did the carotenoid analysis of this. We started to realize that it has some very unusual carotenoids. This is the carotenoid biosynthetic pathway in different cyanobacteria. But we find that when we go from low light to high light, we get an increase in this mixoxanthosol, canthaxanthin, and euchinone, which are all cyanobacterial specific carotenoids, but you actually get somewhat of a reduction in the beta carotene. But when we look at the interdimer domain, we almost get only this mixoxanthosol carotenoid that is released. And so this is an interesting carotenoid in that it's a glycocarotenoid, or it's a carbohydrate containing. It has on one end a galactose group. And the thought is that maybe this carbohydrate group can somewhat mimic MGDG, which is a galactolipid that's intimately associated, because we do not know truly what's inside of this. We just know that the carotenoids are there. So what regulates this tetrameric form? Everyone knows that the tetrameric form, or at least the trimeric form, appears to be associated with the packing or the assembly of one single subunit of PS1. It's the PSAL subunit. And when we looked at these sequences, this is just a few. We've done a lot more in our recent paper, but here's TE. You can see the structure. It has 3 transmembrane spanning domains. And you see that the top 2 are trimer formings, and they have a big gap right here, and they have an extension at their C-terminus. Now, if you look at the monomeric PS1, Chlamydomonas and Arabidopsis, they have a bigger insertion between transmembrane 2 and 3, and even more of a reduction in their C-terminus. And so it looks as if our PSAL gene structure is somewhat intermediate in design between the trimeric species and the exclusively monomeric species. And so when we try to model this, you can see some of the effects that happen largely in these loop regions that are basically somehow going to interfere with the stable assembly of these trimeric bundles, this helical bundle that's going to drive the tetramer. So in collaboration with Egbert Bokema, another serendipity is that I got to know Egbert Bokema because I came here. I was in one room or another at the photosynthesis conference about 7, 10, how long ago was that? 8 years ago that was here? 6. And he told me, basically one of Bob's graduate students told me, you have to go look at this poster because it looks very much like your poster. And so we went over in there. That's when we learned that this group was working on the Nostoc. But we've continued with Egbert, and we finally now are about to publish a 3.9 angstrom cryo-EM structure. And you can really see that the structure has all of the same organizations, except that the way the PSAL interface is designed is quite different. And it turns out that the dimer of dimers has really different interfaces that drive the assembly. And so we get a dimer of dimers. And the PSALs come together very well right here, but they don't touch any of the PSALs on this other side. So these two interfaces are very, very distinct. There seems to be some openings right here that are a little bit unusually large. And so we are pushing the structure analysis of this forward as I speak. Now, to the speculation, I have a few more moments. So we discovered this about 5 years ago, and we speculated that this evolution of photosystem 1, this is the higher plant or algal system over here, where you have a really asymmetric structure. You have PS1, where you have two faces of it. The two faces interact independently with different antennae. They interact with the LHC1 on one side, and then depending upon the conditions and the state transitions, they can interact with the LHC2. So there was this need to sort of break the symmetry so you could have a front and a back or a top or a bottom, whatever. The trimeric system really didn't, and actually was a very good system to endure maybe high thermal tolerance, or in the case of iron stress, accessory pigments for that. But somewhere, something happened with the changes in the sequence, and you created a different structure which now could form either a dimer or a tetramer. Further changes might have happened, and then we had the addition of the PSAH subunit, which essentially caps that PSAL such that it can't oligomerize. And this was kind of our working model until last year, when a new paper came out trying to identify that there's a new cyanobacteria, shown right here, that's an early branching freshwater cyanobacteria that sits basically right at this point here. And so this is all of the cyanobacteria in this particular tree. These are all the clear trimers. These are all the tetramers. These are a little bit of a question mark. And then coming right off of this cyanobacteria were the glycophytes, which are considered to be, by everyone's analysis, a early intermediate in the evolution of plastids. And so we went and we looked at the glycophytes, and it turns out that the glycophytes have also a tetrameric or a dimeric photosystem 1 as well. And so here's T. elongatus and Seneca cystis. Here's Anabeta with a tetramer. Here's two different glycophytes right here, and you either have a tetramer or you have a dimer. So we think maybe, we don't quite know where this transition takes place. I think maybe this model could be turned around. And so if you look at the model that's been put forward, and where did plastids come from, there's really two theories. There's one that they came from some of these heterocyst-forming species over here, either branching or not. So there's one model here. The new paper argues that they probably came from this group over here, that this early branching freshwater cyanobacteria is the real origin. And I think maybe our work suggests, the Glioblastoma is for sure a trimer, is that maybe if we turn this thing around, maybe there's an ancestral, this heterocyst and close relatives that actually maybe could somewhere be a precursor to some of these early cyanobacterial species that are collectively called the Crococales, and that they could have given rise not only to this pathway, but also into the heterocyst and the close relatives. Okay, I just want to stop here. As I said, I was his first student, but I have a picture here of Ben being the last student. And I just want to say thanks. There's been many great people that have come between us, and I'm glad to see so many of you here. And as a final testimony to Bob, I was almost going to name my title, How to Be a Hot Springs Junkie and Call It Work, because in my spare time, I go to hot springs all over the world. Okay, thank you.
Well, thank you very much, and thanks to Joe for inviting me to come. I'm really pleased to participate in this. I thought that the cover of Bob's book, about the second edition of Bob's book, was a good metaphor for this symposium. In a way, it's marking the transition of Bob from the first edition of Bob Blankenship's career to the second edition of Bob Blankenship. And so, as was already mentioned, I think by Govinji, got to know, Sarah and I got to know Bob and Liz during the time that we were postdocs in Seattle in Bill Parsons' lab. And I won't tell you what year that was, but this is Bob dancing with our 2.5-year-old daughter, who is now a lawyer in LA and has a daughter of her own who's about to go to university this fall. So. And so shortly after this photo was taken, both Bob and I left Bill's lab to start our own real jobs. And for both of us, it turned out to be real jobs, an academic career. And, and pretty much we did photosynthesis research for our whole career. But in pretty different areas most of the time. And so Bob did things that, we just heard about from Barry, while I was doing things that were, tended to be more physiological. But one area that our research did overlap, particularly in the last 10 years, has to do with the area of redesigning photosynthesis. And in 2009, Bob organized a meeting in Albuquerque, New Mexico, that was looking at comparing photosynthesis and looking at improving photosynthesis. This is a paper that was published in Science 2 years after that meeting that summarized and expanded on the conversations that we had during that workshop. And so one of the things that this paper did was it tried to make an apples-to-apples comparison of the solar conversion efficiency of solar cells versus natural photosynthesis. And I'm sure Bob wrote this, and he was very optimistically saying that for short terms, photosynthetic conversion can get within 2 to 3 of the efficiency that solar cells can do. And so I think partly because of that, it also inspired during that meeting and in this paper a conversation of, well, how can we get natural photosynthesis a little bit closer to what solar cells do? And of course, solar cells are great for making electricity, and one of the things that we tried to do in this paper was, in the apples-to-apples conversion, make the comparison of making the solar cells make hydrogen from electrolysis. But nevertheless, solar cells are never going to make food, and they're never going to make feed for the animals. And so there is a reason to increase photosynthetic efficiency. And so in this paper, there was some pretty aspirational ideas of how photosynthesis might be re-engineered, and it's typified by this, and that is to try to mimic a 2-junction solar cell in natural photosynthesis. And so the way the natural photosynthesis works, I guess evolution wasn't all that bright in thinking about this, even though it has two solar systems or 2 photosystems. Both of those photosystems use the same region of the spectrum, and therefore it automatically divides the efficiency of natural photosynthesis in 2. And so the aspirational redesign, and I think this was mostly driven by Tom, this idea, Tom Moore. And so this idea was, well, let's see if we can make a biological mimic of a 2-junction photocell. And it turns out that biology had already created both of those systems. And so photosystem 2, from higher plants, it's able to oxidize water that uses the visible spectrum. And then the reaction centers from purple bacteria or other photosynthetic bacteria that use the spectrum out in the infrared. And it turns out that the energetics work out pretty well. And so the overpotentials are big enough to do the kind of reactions that have to occur. And so this could, in fact, if you could implement it, would actually double the photosynthetic conversion efficiency of solar energy, a much bigger spectrum than what plants do now. Of course, from an engineering standpoint, this is pretty daunting. So even though these 2 reaction centers exist in nature, they come from widely divergent organisms, and the notion of being able to actually engineer these 2 things into a common membrane that could undergo water oxidation and also use the energy out here in the infrared was pretty aspirational. But nonetheless, it was fun to think about. Then about 4 years later, Bob participated in another workshop on redesigning photosynthesis that I organized. It was the Banbury Conference at Cold Spring Harbor in 2013. And then 2 years later, we published a paper in PNAS about the ideas that we had for redesign. And many of these also were very aspirational from the standpoint of they were fun to think about, but it was really difficult to believe that there were realistic goals in being able to engineer it. Having said that, one of the things that we talked about at that conference was trying to optimize the size of the photosynthetic antenna to match what would actually be optimal for crop photosynthesis. And so in crop photosynthesis, we have these very, very dense stands of monocultures that absorb virtually all of the light that hits them. And so in Bob's book, he has this wonderful little section that's called Why Antenna? And I always use this when I teach photosynthesis, when I introduce antenna. And what Bob does in about a page and a half of this book is he leads you through this very much conceptually driven back-of-the-envelope calculation that brings you to the conclusion that one chlorophyll molecule at full sunlight will absorb about 1 photon every 1/10 of a second. And this is about 50 times too infrequent to make an efficient photosynthetic system. And so the answer to this question of why antenna is that it takes about 50 chlorophylls in order to justify investing all of the energy that it takes to build the rest of photosynthesis to use those photons. And so, you know, conceptually this really explains why it was necessary to have antenna. But what came out of that conference was another question, and that is, why so much antenna? And the reason for that question is shown here. And so this is the rate of leaf photosynthesis measured by CO2 gas exchange versus the light intensity. And so this is in the visible spectrum, so it's the photosynthetically active photon flux density. And full sunlight is somewhere between 1,500 and 1,800 microeinsteins per meter squared per second during the growing season in this part of the world. And so what this shows, this is for a soybean plant. And so what this shows is for virtually all of our crop plants, and nearly all of our crop plants with the exception of maize, are C3, is photosynthesis saturates at about 20% of full sunlight. And so what that means is that at the top of a soybean canopy or the top of a rice canopy or at the top of a wheat canopy, about 75% of the day is spent with about 4 to 5 times more photons than photosynthesis can actually use. And so not only are they wasted, but they are absorbed, and so photosynthesis and the plant has to deal with them in a way to dissipate them so that they're not destructive to the photosynthetic apparatus and to the plant. The paradox is that, you know, a single green leaf has an optical density across the visible spectrum of about 1. And so it absorbs 90% of the light that's available. And so the paradox in a crop canopy is here at the top, you have way, way more light than you can use, but 90% of that is taken out by the top leaf, so 1 leaf layer down, it's light starved, so it's light limited. 2 leaf layers down, you're down to 1% of full sunlight. And so this is a huge inefficiency in C3 photosynthesis, at least in dense agricultural canopies. And so this got us thinking about, well, if that's the case and the antenna is too big, can we design a smaller antenna so that we use less of the light at the top of the canopy and allow more of the light to penetrate into the canopy where it can be used more efficiently. So we wanted to increase the transparency of the leaf. And, you know, before we sat out on the engineering, we wanted to do a little bit of modeling, and I just want to direct your attention down here. And so this is canopy extinction coefficient, and so we can take this as the surrogate for the chlorophyll concentration. And then this is the amount of photosynthesis that occurs over the course of a day. And so for this model, and so this model is taking account of the fact that the sun and leaf angle changes over the course of the day, as well as the intensity of the sunlight changes over the course of the day. And so we modeled this for 40 degrees north latitude, which is the growing area of the central corn belt of the United States. And we did it for the 200th day of the year. And so that's right in the middle of the growing season. And this is for a soybean canopy, and we modeled it for a situation that has a leaf area index of 3, which means that the leaf area is 3 times the ground area that it covers. And what this simple model suggested is that this is the full green soybean leaf, and this is the integral of carbon gain across the day, and it suggests that if we decrease the canopy extinction coefficient, if we decrease the chlorophyll by 60%, we would see a 25% increase in the daily integral of photosynthesis. This is a simple model, and it's simplified in ways that I'll show you later probably overestimate this, but I'll get to that in a second. And so the biosynthetic pathway of chlorophyll biosynthesis in higher plants is pretty well known. All the players are known. It's shown here on this slide, and where these are located in the chloroplast, there's a little bit of debate, which is why you see some of these repeated. But there are lots of different targets to be, look for down-regulating the biosynthetic pathway to produce less chlorophyll A in the plant. And so we targeted two. We targeted magnesium chelatase and chlorophyll synthase using microRNA and RNAi to down-regulate the amount of chlorophyll that was being made. And this worked. And so we got light green plants. They look like house plants, but they were light green. And so we wanted to look at how this affected light penetration first at the leaf level. And so we used a technique called laser sheet microscopy. And so what we do is we illuminate the top of the leaf with a band of laser light of different color, and then from the side of the leaf we can detect chlorophyll fluorescence. And so the intensity of the fluorescence you get out is proportional to the amount of excitation that penetrated into the leaf. And so so these are just false color images of the fluorescence intensity that's coming out for the full green leaf and for the leaf in which the chlorophyll concentration was reduced by 60%. And so here's the full green leaf, here's the top of the leaf, and so we're shining the laser here, and then we're detecting fluorescence at a 90-degree angle as a function of depth. And here's the leaf in which the chlorophyll has been reduced. And so in the full green leaf, you can see that the blue light, all of the blue light is absorbed very close to the top of this leaf. Red light penetrates a little bit deeper, and then green light penetrates quite deeply into the leaf. And this makes perfect sense. And so, this is what we know about chlorophyll. Very strong oscillator strength in the red and the blue, not very much oscillator strength in the green. You can see then when we have reduced the chlorophyll concentration, blue light's penetrating a little deeper, red light's penetrating deeper. It doesn't affect green light that much because green light's already penetrating pretty deeply. And so we can quantify that. And so when we quantify it, you can see what I just said. Blue light is getting deeper in the leaf, red light's getting deeper in the leaf, and the green light is penetrating somewhat more deeply, but it already penetrated quite deeply. Having said that, just a point about green light is that in dense canopies, in dense full green canopies, about 50% of all photosynthesis is driven by green light. The reason being is that it does penetrate through the leaves and it can get down into the canopy where it can be used more efficiently, where red and blue light don't. And so sometimes we get grant proposals about, shouldn't we shift the green light into one of the stronger absorption bands of chlorophyll? And we would say, no, you don't want to do that. So, Gavinji always asks me why leaves aren't black, and that's the reason they're not black. And so when we do that, and then we look at leaf photosynthesis as a function of absorbed photons, photosynthetically active photons, you can see that the light green leaves outperform the darker leaves, even at the leaf level. And so even in a leaf, you have a gradient where the chloroplasts at the top of the leaf are much more saturated than the chloroplast at the bottom of the leaf. And so if you can allow more of that light deeper into the leaf, you do get an increase in efficiency, at least on an absorbed photon basis. Now, I said that original model of ours was overly simplified. And the reason it's overly simplified is one of the things we all know is that there's 3 fates for a photon that hits a leaf. It's either going to be absorbed, it's going to be transmitted, or it's going to be reflected. And so what we didn't account for in that original model was how much of that light was reflected. And the reason that that's important is any light that's reflected at the top of the canopy has a very high probability of just being reflected back into the atmosphere and being lost from the canopy. And so this suggests that there's going to be some sweet spot between the light that's lost due to reflection versus the improvement in photosynthetic performance due to greater transmittance to deeper into the canopy. And so there aren't any really good ways to model this. And so the approach that we took at the University of Illinois, we have the soybean germplasm collection for the United States. And in that collection are 68 light green soybean mutants. Most of them are light green for really bad reasons. You know, they don't metabolize nitrogen properly or whatever. And so most of them are really, really unhealthy plants. But they're healthy enough that we could grow them out into the field and determine the optical properties as a function of chlorophyll concentration in a single species, in soybean. And so we grew out these 67 species and we determined the optical properties of them in soybean. And the chlorophyll concentration varied from about 100 micromoles per meter squared of leaf area to full green, which is about 500 micromoles of chlorophyll per leaf area. And so we had a nice big variation in chlorophyll concentration. And what we found, not too unsurprisingly, is that leaf transmittance and leaf reflectance increase with a decreasing chlorophyll. So you can see that here. Here's leaf reflectance. Chlorophyll is going down and it's going up. In the case of leaf transmittance, which is what we want, that's also going up as chlorophyll concentration goes down. And then leaf absorption, of course, has the opposite, and that is as it goes down, or as chlorophyll goes down, so does absorbance. What came out of this that was really important for our modeling is that the ratio of reflected light to transmitted light always stayed about 1 regardless of chlorophyll concentration. And so earlier models had suggested that, that they did change as a function of chlorophyll concentration, but at least when we did this empirically in soybean, it doesn't. And this has important effects on the model because, you know, previously, if we had used the empirical models that are out there to model this, that had transmittance going up more rapidly than reflectance. And so we took this information and we parameterized it into a multi-layer canopy model. And so multi-layer means that we're looking at 10 different equally spaced layers within the canopy, and then modeling what the chlorophyll concentration and what the light intensity would be as a function of the time of day and as a function of the time of year for a full green canopy as well as a canopy in which we had reduced chlorophyll. And so what you can see here is that, and so this is day of the year, and so this is somewhere about 1st of June here, and this is the end of the growing season out here. And what's going on here is we're just comparing the difference between a full green and this light green. And so you can see during the beginning of the growing season, there's really a pretty big penalty for being light green in terms of absorbed photons. And so at the top of the canopy, we're losing a lot of photons. A lot of that is being lost to reflectance. And there's no advantage of being light green early in the season. You only have one leaf layer out there. And so if the leaf is transmitting it, it's just going to transmit it to the ground that doesn't, you know, and that doesn't benefit the crop at all. Later in the season, you can see that now there's some benefit to being light green. We're losing some at the top of the canopy due to reflection, but now we are getting an increase in light absorbance deeper in the canopy. And then this is at the end of the season during senescence. And then this is canopy gain. And so this is the daily integral of canopy gain as a function of day of year and height in the canopy. And you can see, unsurprisingly, that it pretty well mimics the absorption profile. And so the result of that, when we put it into the model, is that there is a small depression in the amount of light that's being absorbed, and there's a small depression in the daily integral of photosynthesis. But this is a reduction of 70% of the chlorophyll concentration in the canopy. And so there's a huge amount of nitrogen savings that are associated with, not just from the chlorophyll that you're not making, but more importantly, the chlorophyll proteins that you're not making. And so the nitrogen use efficiency in these light green plants could be as much as 60% higher than what it would be in wild type. There's another advantage that might not occur to you, and that is that one of the aspects of photosynthesis is that it takes a lot of water to fix a molecule of CO2. So under normal growing conditions for a soybean plant, for every CO2 molecule that's taken up, it costs about 700 molecules of water being lost. And so this is driven, of course, just by the fact inside the leaf the atmosphere is saturated with water, and so there's a driving force for transpiration into the drier atmosphere. If you do photosynthesis deeper in the canopy, that driving force, the vapor pressure deficit, is lower. And so if we're doing more of the photosynthesis down in here, our model suggests that there should be a substantial increase in water use efficiency. So just briefly, I want to talk about one other strategy that we use for increasing the transparency of the leaf. That was to change chloroplast size in order to maximize absorption flattening or the sieve effect. I always explain the sieve effect in photosynthesis class with the notion of taking an ink bottle and putting it in a bathtub. So if you take an ink bottle and you put it in a bathtub, and it changes the absorption of that bathtub very, very little. If you divide that same amount of ink up into 1,000 little bottles and you put it in the bathtub, it changes it more. You'll get the maximum increase in absorbance if you just pour all the ink into the bathtub. And so the analogy here is that if we can make chloroplast size larger, not change the total chloroplast volume, and stuff the same amount of chlorophyll into fewer compartments, then we'll maximize absorption flattening and increase the transmittance of the leaf. The advantage of this, in principle, is that it shouldn't increase reflectance. And so the way to do this is Kathy Osteryoung's lab at Michigan State showed some years ago in Arabidopsis that you can titrate FITZ1 and FITZ2 that are involved in chloroplast division. And so if you titrate the amount of this protein that's being made with RNAi or microRNA, you just get progressively larger chloroplasts. And so we've done that. This is done in energy grasses because this happened to be supported by the DOE Foundation, and they don't care about soybeans. And so we're doing this in sugarcane. And what this shows is that here's wild-type sugarcane, a chloroplast volume about 34 cubic microliters. And here we've titrated it with FITZ to make larger chloroplasts, and they're 5 to 6 times larger. We've also done that in sugarcane, or in sorghum. And here is the wild-type sorghum, and this is looking again at light sheet microscopy. So we're looking at the depth of penetration of these different colors of light into the leaf. So here's the parental full green. Here's a light green in which we've increased the chloroplast size by about a factor of 10 in this case for sorghum. And what you can see is that there is deeper penetration into the leaf. And so this does increase leaf transparency, but it's pretty small. And so the biggest that we're able to see is about a 10% increase in transparency. But when we look at the optical properties of the leaf, it does not increase reflection at least in our ability to detect it. And so what are the next steps in this project? I think one's obvious, and that is to combine the low chlorophyll with the larger chloroplast trait. And so our models suggest that this could be the ideal situation of really finding the best sweet spot of lower chlorophyll and minimizing the amount of light that's lost to be absorbed by the canopy. The other thing I mentioned was that there's a real disadvantage to being light green early in the season, and so we're looking at inducible promoters to be able to turn on our RNAis and shut down or tune back chlorophyll synthesis after a leaf layer is formed across the entire field. And so right now we're using just ethanol inducible promoters, and so I have a postdoc who's out in the field right now spraying a lot of tobacco plants with ethanol. But later on, we want to do it with light-inducible ones. And so one of the things that happens as you absorb light through the canopy is you change the red to far red ratio. And that's what the photomorphogenic signal phytochrome responds to. And so there are promoters that we can use that will respond to that change in color ratio. And so that's the eventual plan. This has two potential advantages. One is what I've already stated, and that is to try to minimize the time when they're light green, when it's a disadvantage to being light green. But the second thing is that the model suggests what you'd really like once you form a canopy is to have light green at the top of the canopy, and then as you go deeper into the canopy, you'd like to get fuller green and fuller green because light intensity is dropping. And so again, using these phytochrome inducible promoters or phytochrome sensitive promoters, we may be able to do that. And then finally, another way to get around this reflection problem is to make leaves more transparent by making them thinner, and making them thinner by having fewer leaf layers. And so having fewer cells that retain chloroplast. And we have a few mutants that have fewer leaves, or have fewer cells in the leaf layer, and they do seem to show this trait that we expect. We're trying to find some tractable genetic controls to be able to do this in a patterned way so that we can look at this more systematically. And so that's what I have to say, and here's a more modern picture of Bob and Liz and Sarah and I. Thanks very much. Yep. Well, I mean, essentially that's what we're doing when we're enlarging chloroplasts, is reducing the number, but we are increasing the volume. And so we haven't changed the total chloroplast volume, but we have changed the number of chloroplasts. And so we don't know how to just change the number of chloroplasts that have the same size. But that would be another way to go about it if we did. Yes? Yeah, and so the question about chloroplast movement, we've looked at that a little bit, and they still move. They appear to move more slowly. We're not particularly concerned about that in the sense that no one has ever really shown that chloroplast movement has very much consequence to photosynthesis. There's one school of thought that it's photoprotective, and so they stack up at the side of the cell and photoprotect the ones that are below. There's another school of thought that, in fact, it doesn't have anything to do with photoprotection. It has to do with high light. The chloroplasts want to get as close to the source of CO2 as possible, and so they stack up next to the intercellular airspace. And so years and years ago, we looked at this in Arabidopsis to try to see if we could figure out in mutants that couldn't move chloroplasts versus ones that could, if we could see a photosynthetic consequence for that. And we were never able to resolve one. Your second question about ultrastructure is, we've done this in C4 plants, and it's complicated because there are two kinds of chloroplasts. There's the mesophyll chloroplast and the bundle sheath chloroplast. And we are doing the microscopy right now to try to sort that out, and we just don't know at this point. Uh-huh. Yes? Yeah, and so this isn't work we're doing, but it's funded on the same project of looking at the effect of leaf angle in soybean and in some of the grasses. And in the grasses, it's pretty clear. And so in maize, for instance, if you look at maize today versus maize 50 years ago, they have a much, much more erect leaf angle. And all the models suggest that's much better for light penetration in the canopy. What's complicated this, however, is that at the same time that breeders have selected for this more upright angle, they've also selected for crowning tolerance. And so what's happened is that along with this upright structure, planting density has gone up. And so planting density has gone from 30,000 plants per acre to 54,000 plants per acre. The last 2 decades of yield increases in maize is almost totally due, to, this increase in planting density, allowed in part because of this more upright structure. Plants like soybeans, it's more complicated. And so soybeans originally were periheliotropic, and so they track the sun, and most of that's been bred out. But we have been looking, in my group, we've been looking at the effect of shorter petioles, and so there is a genetic control over petiole, and modeling suggests that, to get this Christmas tree architecture in soybean plants. And so that works in models. You put these plants in the field, and they keep the Christmas tree architecture until canopy starts to form, and then they lose it. And so that particular trait turns out not to be very useful. But you're absolutely correct that leaf architecture is huge in terms of light distribution in the canopy. Barry?
So, 2 questions. Does this explain why in a garden setting, my tomato plants, I put a red cloth around the base of a larger plant? Are you familiar with this?
I'm not familiar with it, but I can answer your question. No, it doesn't explain that at all.
I've done the experiment. It's absolutely an increased safe plant with a reflected red polymer around the base.
Oh, if it's reflective, then yes. And so if it's reflective, then yeah. And so this, does it have to be red? Yeah. And so this is done a lot in grapes too, where they line the surface, and it does reflect light back up into the canopy and illuminate some of those leaves that otherwise would not be illuminated. And so yes, so I would say that it does. It does.
The other question is, how do Grana stacks affect this? If you were to get a lower stacking view, wouldn't that give you somewhat the same effect?
Not if you didn't change chlorophyll concentration. And so, and we've not looked at some of the barley beetless mutants. Of course, the barley beetless mutants are light green, and so it would be hard to differentiate those. But I think the modeling would suggest that it's going to depend on, there's going to be more shading in those stacks, and so it might make it worse. We've not modeled that.
Low light plants tend to have higher stacks.
Yeah. And so, in Bob's calculation about antenna size, that was for full sunlight. And one of the things he points out is that's the best situation. And so as you get to lower and lower light, you need more and more antenna. And that's exactly what these low light plants do. And in fact, that's what shaded leaves and highlight plants do. So they also get bigger antenna. Yeah.
All this sounds very promising. What are the prospects of adopting it for the hundreds of square miles of farmland?
Yeah, and so, that's a difficult question. And so we have shown proof of concept in soybean that it can be done. I think there's a couple of cultural things to think about. One is that probably the reason it's never been selected before as a trait by breeders is breeders see anything that's light green, they throw it out. And usually with good reason, because they're usually light green for really bad reasons. Every now and then there'll be a mutation in chlorophyll synthesis. And the second thing is that farmers hate light green, right? For the same kinds of reasons. And so I think there's 2 cultural things in there. And then the third one is that one of the things that is important about dense planting, and they do it, is weed control. And so if you make them light green, particularly early in the season, then you're not going to get the kind of weed control that they're used to, and you probably have to apply more chemicals. Another reason that we're thinking about the inducible promoter is to keep them green early in the season.
Well, we're just about out of time.
All right.
But thank you very much. It's really interesting.
So before we take our break, we're all here because of Bob, but there's somebody who actually organized everything, and that's Amanda, who's back here at the door. Give her a round of applause.
Thank you very much, and I really want to thank Joe and others that were involved in putting this together and inviting me to give a talk at this celebration of Bob's scientific career, which is really I couldn't miss it. This is a great thing to do. So the title with this, Traveling with Bob Through Early Earth Photosynthetic History, I'm just going to talk about common threads. And so we have a number of common threads that Bob and I interacted many times over the year. We even had a common grant once to sequence a lot of these photosynthetic organisms. So what is going to be our various different things that we traveled together and had very common interests in the origin and evolution of photosynthesis. Let's see if this is it. Okay, so one of the common threads, the early threads that I had was with Bob, has to do with Howard Guest. So when I came to Indiana University as a young little squirt, where you can see over on the right there, that's actually me, pre-beard. That was actually in 1988. And when I went to Indiana University, Howard had just retired. And I was a new young molecular biologist who did a lot of cloning and sequencing, which to Howard was mysterious. But I was not a microbial physiologist. And so when he found out that I was working with photosynthetic bacteria, he immediately says, well, you have a startup package? I said, yes. He says, well, we need a really good microscope. And I said, but I've never used a microscope, because all my research was on, lambda genetics and phage lambda. And so he worked with me for about 2 years before he finally got his microscope out of my startup package. But the reason I put this up here is that one of the things that Howard loved to do was to take me out in the field to show me where photosynthetic bacteria lived. And that really perked my interest about the origin and evolution of photosynthesis and where it came from. But the other thing was that Howard was really an interesting guy, a really interesting character, and he loved to do travel, loved to talk to people, loved to interact with other scientists, but he hated Indiana winters. And so one of the things he liked to do in the winter was to get out of Indiana and go to California or go to Arizona and visit the other scientists who worked photosynthetic bacteria so he'd have a warm place to go. And so frequently he would go to Arizona and he would camp out at Liz and Bob's place and make himself at home, and he would come back from those visits and tell me all about the great research that Bob was doing in his lab. And one of the stories he told me, and I didn't believe it at the time, was that Bob actually grew these photosynthetic bacteria in cabinets. It was like literally a cabinet in the lab. You open up the door, and he had a naked bulb in there, and that he would adjust the temperature by opening and closing the doors. And I came to Arizona in I think it was in 1992, and I went to Bob's lab, and by God, he was growing bacteria in cabinets with a bulb and by opening and closing the doors to control the temperature. So that was one of the early things I had with learning about Bob through Howard. Okay, so the other thing that where we interacted was on this, I call it the New Testament of the photosynthetic bacteria Bible. So the first testament was Clayton and Systrome, who published this phenomenal treaty in 1978, I think was when the publication of that was. And that was the photosynthetic bacteria book that everybody used when I was entering this field. It had all the information on the physiology and the biochemistry of these organisms. But at that time, there was before the cloning, it was before the crystallography, so there wasn't any of the more modern stuff. And so at some point, I don't know if it was Bob or who it was, or Gavinji, but Gavinji had this series he was starting, which was the photosynthetic book series, Advances in Photosynthesis. And so this is actually book number 2 in that beautiful series of about more than 15 books, chapters in here. And Bob called me up and invited me to become a co-editor of this book, and I was shocked because I was such a young squirt in this field. But the reason I put this up here is that you can tell by the book, this is the copy, the version in my lab, these things all get beaten up. And the reason they get beaten up is because it has so much information. And if you look at Bob's Google Scholar, you'll see that this is actually one of his more cited works because it was used for many, many, many years by people who work in this area. And the other thing is that this book had 3 different sections. It was more of the structural, the crystallography of reaction centers had just come out, and all these biophysicists poured into this field, and they're all arguing as to whether or not it's 10 picoseconds or 5 picoseconds that these electrons are moving around from these components. And so Bob was right amongst that middle of that battle. It was a really exciting time in reaction centers and light harvesting and how they work at a molecular level. But also at the same time was when sequencing occurred, and all of a sudden we had all this primary sequence information on all the structural polypeptides and on the chlorophyll biosynthesis genes. And then we also had all these different new forms of photosynthetic bacteria that are coming out. And so we had these different areas. I was a molecular biologist, so I did the sequencing. Bob did all the biochemistry and biophysics, and Mike did the physiology. But the reason I put this book up here is that Bob is so unique. He could have done the whole editing of the whole book. Absolutely. It would have killed him time-wise, but he knows all those areas, and he's one of those few people that can wear these different hats. So he's equally as home in the biology department with the microbiologists as he is in the chemistry department with the biophysicists, where he's talking about how those electrons are moving from the one chlorophyll to the next chlorophyll, how to measure that, and the importance of that. So it's really very unique that he has this breadth in this area. Okay. So what I'm going to talk about are the common interests that Bob and I have over the origin and evolution of photosynthesis. And so it's really, photosynthesis, everybody, I don't have to say in this room, but photosynthesis is the primary way that light energy is absorbed and converted into cellular energy. It's an amazing phenomena. If we didn't have photosynthesis, we would not have Earth, this beautiful evidence of life on Earth like we can see from outer space, and we also wouldn't have any really significant amounts of oxygen. If we didn't have photosynthesis, this is actually from the Mars rover, it would be more like what you'd see in a place like Mars, where you're not going to support much biomass. There may be life there, but it's not going to be the amount of biomass like you would have on Earth because of the primary absorption of light that is used in photosynthesis that captures that energy and uses it for cellular energy, which is used for the primary food chain. So really, if you think about it, Earth, the nature of Earth changed rather dramatically when photosynthesis evolved. The other thing to think about is that photosynthesis is the most polluting thing that's ever happened on Earth. Prior to photosynthesis, there was no significant amounts of oxygen. Earth was very reducing. But upon the evolution of oxygenic photosynthesis, all of a sudden you have all this oxygen pouring into the atmosphere, which changed the composition of the atmosphere to make it more oxidizing. And that had a very profound impact on life as we know it. So one of the questions that I've became interested in, and this is a question that Bob was interested in for many years, is how old is photosynthesis? And the answer to that is very, very old. It goes much older than Bob or I. It goes way back in the evolutionary history of Earth. And we can tell that through several ways. One thing that you can tell is that you can look at oxygen. So this is the amount of oxygen that's present on Earth. This is based on geochemistry versus time. And so here's current modern day versus 1 billion years, 2, 3, and 4 billion years ago. And you can see these bursts of production of oxygen. Well, the only way you're getting oxygen on Earth is through photosynthesis. And so what that's telling you is that oxygenic photosynthesis really dates itself out here around 2.8 or so billion years ago. So it really, really is an old evolutionary process. You can tell this information based on these iron bandings. So this is actually, I took this picture when I was in Arizona and I was driving along because it shows you this beautiful iron band that occurred here. And the reason you have that iron band is that when the Earth was reducing, iron was in a reduced state in the oceans and it's very, very soluble. But when iron becomes oxidized or rusts, it's very insoluble. And so there was this catastrophic event when oxygenic photosynthesis occurred. You made oxygen, the iron started rusting, and it rained out of the oceans. And so not only were cells challenged with the presence of an oxidizing atmosphere, they're also challenged with a limitation in iron that was occurring because of the raining of the iron down, but it also really well allows you to date where photosynthesis occurred just based on this iron banding. Okay. Oxygenic photosynthesis we can date around here because of the production of oxygen. And so one of the things I want to argue, and it is still to this day a matter of discussion, is whether or not the simple photosystem we see in the anoxygenic photosynthetic organisms predate the complex photosystem that you would see in cyanobacteria and chloroplasts. And if that is indeed the case, then that pushes the actual origin of photosynthesis much deeper into the origin of life on Earth, probably at least 3.5 billion years ago. So to understand this a little bit, I just want to just introduce, probably don't have to say too much about this in this audience, but there are different types of photosystem. So we have a photosystem 2-like photosystem that's present in the purple bacteria and in the green gliding bacteria. It's a very simple photosystem, unlike photosystem 2 in plants, which have many, many peripheral additional polypeptides that are there, which are used in water splitting and other functions. We really only have 3 different proteins at this reaction center, and those 3 proteins combine chlorophyll and carotenoids and quinones and have a very efficient process where they reduce the quinones that goes through the BC1 cycle and then back to the reaction center. So it's cyclic electron transfer flow, and the ultimate thing that happens is you're moving a proton from one side of the membrane to the next, and then that proton gradient is used to generate ATP through ATPase. So basically they're just pumping electrons around in a circle. In the PS1 type, there's a heliobacteria, and then there's a green sulfur bacteria. The heliobacteria is a gram-positive. The green sulfur bacteria is these organisms that can also use sulfur as electron donors, but they also go through typical cyclic electron transfer and generate a membrane potential, which can be used to generate energy. These have a PS1 type reaction center. Chloroplasts and cyanobacteria have coupled these 2 together, so you have the PS2 and you have the PS1. Cyclic electron transfer, the most important part of this is the water splitting, where you're taking electrons from water, generating oxygen as a product. That is your oxygen production on Earth for the last 2.8 billion years, and then you generate reducing power as a product. So this actually was some of the very first work that I saw from Bob back in the early 1990s, and even earlier. We saw a talk earlier today. Bob has been interested in the origin and evolution of photosynthesis forever. And actually, I took this slide from an early talk that I probably gave about 10 or 15 years ago, where I used Bob's slide in one of my talks, because this actually is some of the main questions that both Bob and I are interested in, which is in current organisms today, we have the green and the heliobacteria, which have PS1, we have the purple bacteria, which have a PS2, and the cyanobacteria that's coupled. And there's several models how that could happen. Did we have a PS1-like organism and a PS2-like organism, and those 2 photosystems coupled together? Or some of the other models are that cyanobacteria actually represent the most ancient, and that from the cyanobacteria gave rise to the heliobacteria and the purple and green non-sulfur. So there's a lot of debate about where these things came from. And then the ultimate question is, where is the first photosystem occur, and how did those give rise to these complex photosystems we see today? The other thing I want to point out is that heliobacteria, Bob demonstrated very early on in the late 1980s and early 1990s, when he started working with the heliobacteria that Howard Guest isolated, and he demonstrated it had a PS1-like photosystem, but more importantly at that time, he proposed that the PS1-like photosystem was a homopolypeptide. And at that time, that was a big deal, because all up to that point, the PS1 had two proteins involved in forming the PS2 core, and he demonstrated there was only one protein that's there. And it was somewhat controversial, probably, when he first proposed it, but it turns out that he was absolutely correct. So one of the things that my group and Bob's group has been doing for a long time is looking at these different photosynthetic organisms in the bacterial tree. And this is actually a tree from one of Bob's papers. And so what you see here is in the purple, or in the bacterial lineage, what we have are organisms that only have PS1, they're in the green, or only have PS2-like reaction centers there in the purple, or have both PS1 and PS2. And so what you can see is that the archaea have no tetrapyrrole-based photosynthesis, so they're not involved in the origin and evolution of photosynthesis. But the other thing that's important to point out is that the cyanobacteria lineage, if you look at where chloroplasts key out, chloroplasts don't key out here, chloroplasts key out as a branch of the cyanobacteria. And so this actually supports the symbiotic work that was done many years ago, showing that chloroplasts are essentially a cyanobacteria that was swallowed up by a eukaryote, and then that gave rise to the lineage of the chloroplasts that we see in higher plants. So one of the things that we decided to do early on was to go ahead and pull out the photosynthesis genes from each of these branches, and from that work then build phylogenetic trees. In order to do that, we wanted to get each of these different organisms growing in the laboratory and get the DNA from them. And I'm just going to show you a few pictures for this just so people can see where these guys live. When I go to a place like Yellowstone, this is what I geek out on. So we've got a thermophilic area over here, and you've got these beautiful layers of photosynthetic bacteria that are there, and I'm sure Bob's done the same thing. When you typically go to Yellowstone, most of the people, they go there and they see these animals, and they'll stop their car and say, wow, there's a bear, and oh, there's a buffalo. But what I do is I tell my wife, stop. We gotta go to this hot spring, you know? And we'll go to these hot springs, and I'm taking all these pictures, and I'm like, oh God, look at that, it's chloroflexus, and there's chlorobium, there's heliobacillus, this is unbelievable. And so it's just, I sit there and I totally geek out over the photosynthetic bacteria blooms that are out there, which I'm sure Bob does the same thing. If you work with these organisms and see them in their native state, you're like, oh my God, this is unbelievable how pretty these are and the complexity we're seeing there. Anyways, these are where we get these initial cultures of these organisms that have been passed through laboratories all the time. So anyways, we went ahead and we cloned and sequenced and characterized these. So one of the first things we did was to pull out all the genes from Heliobacillus mobilis, which is the gram-positive, which was actually very nice at that time, and demonstrated that indeed it has a single reaction center gene, which actually supported Bob's biochemistry very well, that the Heliobacillus have a homodimeric PS1-like reaction center. So that was really nice to see that the biochemistry supported the molecular biology there. Then the next thing we did is we went to Chlorobium tepidum and chloroflexus. So we got the green gliding and the green sulfur. We pulled out all the chlorophyll biosynthesis genes. Should say this work was published early 1990s, so we were actually cloning each gene one at a time and sequencing with the old sequencing gels manually. So these things, you know, was nowadays you could do this in 2 days, 2 hours probably. It took 2 years, you know, to actually slug through each of these genes one at a time using those old great big sequencing gels. Okay, building these phylogenetic trees, we were able to look at the chlorophyll biosynthetic pathway and look at the origin. And so what we were able to demonstrate was that based on the chlorophyll biosynthetic tree, the most ancient one was the purple bacteria, followed by the green sulfur and green gliding bacteria, the gram-positive Heliobacillus, then the cyanobacteria and the plants. And so we proposed at that time that you can have some models, we could build some models where you have purple bacteria and then you have this green sulfur bacteria coming around and ultimately giving rise to the cyanobacteria like more complex photosystem. There are some other interpretations that can be done, but the phylogeny of the chlorophyll biosynthesis genes actually have held up, I think, pretty well. Bob, however, at the time, I think Bob had a grant with Mike Madigan where we went ahead and sequenced some of these photosynthetic bacteria, the entire genomes, but Bob took it much further. So Bob has actually been working on the evolution of photosynthesis now for almost 30 years, or at least. And so one of the things he did really well, and it was really, I thought, very significant amount of work. We saw from one of the earlier talks from the science paper, I think it was Govinji showed one of the photos. But Bob took a look at each of the whole chromosomes and did phylogenetic analysis, not just of the chlorophyll genes, but a whole bunch of analysis of the various reaction centers and light harvesting, the chlorophyll biosynthesis, but other components. And from this analysis, what Bob was able to demonstrate rather significantly is that each of these various components for the photosystem have their own phylogeny. So what you have are the photosynthetic pigments have a certain phylogenetic tree. The reaction center will have their own phylogenetic tree, and then the antennas have those, but the photosynthetic cells can be actually a chimera of these various phylogenies. So the photosynthetic organisms will grab different units. But most recently, this is actually 2017, his group used insertion deletion analysis to show that indeed the cyanobacteria are probably the most recent, the most ancient, are most likely versions of the gram-positives and gram-negatives. And I'm running out of time, so I just want to show just very quickly. Chlorosomes, Bob's done a lot of work on the chlorosomes, which is the light heaviest in the antenna. And then we have FMO protein, which absorbs the energy from the rods and funnels that to the reaction center. He's done beautiful research in that area. And the only reason I'm showing you that is because of this. This is French Lick Hotel in French Lick, Indiana, a small little town in Indiana. It only has 2 stoplights. The claim to fame of French Lick Hotel is that's the home of Larry Bird, who was the big basketball star before Michael Jordan. But more importantly, Al Capone used to have part ownership of this, so after he got in trouble in Chicago, he'd come down here and he would cool off in this area. But behind there is a gazebo, and the gazebo has a well, and people used to go there in the 1800s and drink the water because it is smelly sulfur water and it gave you diarrhea for 3 days and it would clear you out. And they felt that would get rid of their ailments. And here's Jeff Favinger. You know Jeff. Jeff was actually drinking the water to prove whether or not it worked, and he was sick for almost a week. It was unbelievable. But you go in there way down. You got to drop down a flashlight, and way down in that well is loaded with green sulfur bacteria. And it's because of those chlorosomes. And the reason I showed that is Bob also did a really interesting work with Tom Beatty, where they went to the bottom of the ocean, 2 miles down, and where there's these smoker vents where you have black body irradiation. And what do they find? These organisms. And they're presumably using black body irradiation 2 miles down, okay, wavelengths way beyond, in the infrared, to grow. And I think the generation time they came up with was a doubling per year, something ridiculous like that, longer than that. But the diversity of the work that Bob does is rather significant. And then just last 2 slides. This is what Earth would have looked like for billions of years. You wouldn't have had it in the trees in the background, but you'd have beautiful blooms of purple bacteria, and you'd have these hypothermal vents, which are loaded with these biofilms, loaded with photosynthetic organisms. This is what Earth looked like for at least 2 or 3 billion years ago, loaded with these photosynthetic organisms. With that, I'll stop. Jin Ziyong was the person who did all the evolution in my work, but really, Bob has taken that well beyond what Jin did, and his excellent students and postdocs really hammered a lot of the information we know about the origin and evolution of photosynthesis. So I'll stop.
Thank you. So up next we have Gary Brudvik, who's going to give a remote seminar. And he's a professor at Yale, and hopefully all the AV works correctly and this can sort of take care of itself. Gary?
Yeah, okay, I'm here. Can everyone hear me?
Loud and clear.
Can you, is my screen showing and can you hear me? Okay, well, Bob, I want to start off by congratulating you on your career and wishing you all the best for your retirement. I'm really sorry I couldn't be there. Just came down with a cold, and I decided it was not a time I could travel. But I'm really glad I'm able to connect using Zoom. So I thought we've heard a lot about various connections Bob has had over the years. One we haven't heard yet is his impact on photosynthesis by initiating several very important meetings for the field. And I'd like to talk a little bit about how the Eastern Regional Photosynthesis meeting started. And Bob's, was the founding organizer of that meeting. He actually went on afterwards to organize the Western Regional meeting. I don't think we have anyone who will say too much about that, but that's had an equally big impact on the West Coast. And so I say a little bit about the beginning of the Eastern Regional meeting, and then I'd like to give one short story of a project that actually grew out of the collaborations that were created by meeting all the people on the East Coast and starting to work together. And so going back, so Bob was at Amherst College when I first met him. And I think I first met you, Bob, at the Gordon Conference at Kimball Union Academy in 1981, but it might have been a little before that. I can't remember. But when Bob found out that I was moving to Yale, he thought, well, we should get together. And Harry Frank, who's also a Ken Sauer alum at University of Connecticut, and we've got Bob at Amherst College, and I started at Yale. We should start having some joint group meetings. So we did that, and in the around 1983, and about the same time, Bob had started some discussions. Saying that, well, maybe there's a growing group of photosynthesis researchers in the Northeast. Maybe there's enough interest to create a Eastern regional meeting that would be modeled after the Midwest meeting that had started a few years earlier. And so that was my start with Bob. We were organizing these meetings. We drove up to Amherst, and Bob hosted a joint group meeting, and they came down to Yale. So that sort of got things a little bit organized on the East. And at about that time, Bob sent out this letter. I had a copy of it in my file. And he said, for the last several years, photosynthesis researchers in the Midwest have organized a very successful regional conference. And he said he wanted to do this in the East Coast. His ideal site would be Woods Hole on Cape Cod. And in fact, that was where we held the first meeting, and we've been having it every year since at the same site. And he said, one of the things that's kind of interesting, we'll attempt to keep the cost as low as possible, hopefully $50 to $75 per person. Yeah, it's changed a bit. We're more than an order of magnitude more than that now, but that was how we got started. And this is the cover and the program of the very first meeting. I had a copy of that in my file. And nowhere in it does it say anything about Bob. He didn't even mention that he was the organizer. There was no introduction or anything. It was just all the program with all the abstracts and the schedule. The way that this was started, that we had some discussions at a Gordon conference, I believe, about how we might do this. And Bob said, there seems to be interest. He'll get the first one going. And he got me to promise to do the second one if assuming there was interest to continue it. And Harry Frank agreed to do the third one. So we didn't have any democracy in those days. This was a dictatorship, I guess, where the first three meetings were predetermined. And after the third meeting, during the third meeting, we had an election for future chair, which there was, and that's been the process ever since. So Bob organized the first meeting in April of 1984. And one of the things this meeting has done is it's nucleated people in the Northeast to get together. And this has created a lot of good collaborations. I just wanted to mention one that has grown out of the Eastern Regional Meeting. This is a collaboration that involves both experiment and theoretical work on photosynthesis. And so there's Marilyn Gunner and myself, I think, have been kind of the ringleaders, but then Victor Batista and Doug Bruce and Sergei Vasiliev and all the other group members. This was from 10 years ago at one of the Eastern Regional Meetings that were sitting out on, I guess it's Oyster Bay in Woods Hole there. And so we've been working together now for, I guess, probably about 15 or more years. And one of the projects that I'll say a little bit about has to do with the role of chloride in photosystem 2. And so I was interested to see that the title that I was given for my talk was not one I chose. It was green chemistry. I guess that could mean green photosynthetic organisms. But it's also interesting. I was going to talk about chloride, and chloride, if you have a molecular model set, is usually depicted in green. So maybe green can be the role of chloride. So this has been kind of an interesting story that chloride is a required cofactor for water oxidation in photosystem 2. It's kind of interesting from a chemical perspective that chloride is actually easier to oxidize than water. So why would you want to have any chloride nearby when it could potentially short circuit the reaction? But it's in fact absolutely required. And early work had suggested that it might be a ligand to one of the metals. This was work from Charlie Oakham's group, where he found that ammonia and chloride were competitive. Ammonia was a competitive inhibitor with chloride. And that the only common aspect seemed to be that they were both potential ligands to metals. Both were Lewis bases. And so it was suggested that chloride was a ligand to one of the metals and ammonia would substitute, and that's how it would inhibit. But when the crystal structure came out, it was found that chloride is in fact not a ligand. It's nearby the manganese cluster, but it's not a ligand. And it raised the question of, what's it doing there? How does it play a role? Why is it essential? And so thinking about this, we were looking at, well, it's just right at the top of a bunch of these hydrogen bonding channels, and maybe it's playing a role in protons and release. And so these were modeled, but it didn't really explain how could that play a role, and why would it be necessary. So that led us to think about, where is it? So it's sitting in the second shell. It's forming an ion pair with a lysine residue, and it's right nearby this aspartic 61 residue, which had been hypothesized to be the start of a proton channel. And so how could it be playing a role there? To think about that, we looked at the role of chloride in other enzymes that are activated by chloride, and we wrote a little review article in biochemistry on that. It turns out there are a number of enzymes that require chloride, and one of them is alpha amylase. And there are crystal structures both with chloride and without. And what you see in the presence of chloride is that chloride forms ion pair with a lysine residue, or the arginine residue. And when you remove chloride, a carboxylic acid that's in the active site moves to form an ion pair to replace the negative charge that's missing from chloride. And this is an essential carboxylic acid in the active site. So now it's moved away and it's no longer active. And similarly, angiotensin converting enzyme, where a chloride is ion paired to an arginine residue. When you remove it, this arginine moves away, creates a binding site that no longer is compatible with binding the substrate. So that led us to propose that what chloride was doing was forming an ion pair with this lysine residue, and that when chloride was removed, this lysine would move to form a ion pair with the aspartic 61 and thereby block this proton channel, which had been hypothesized. This is a picture from Jim Barber's paper in Science, where the proton channel was proposed to start at this aspartic 61 residue. So that was the hypothesis. So one of the first things we did, and this grew out of the Eastern Regional Meeting, was to do some computational modeling. So this was involving molecular dynamics simulations that were done by Sergei Vasiliev, also using QMMM models that Ivan Revolta had generated. And these simulations were done both in the presence of chloride, in which case what we see are a bunch of water molecules that form this network that extend from the manganese cluster all the way past the aspartic 61. But when you remove chloride, this lysine residue moves over to form an ion pair, just like we hypothesized, and it breaks this connection of water molecules. So it seemed to be consistent with the idea that this is a pathway for proton transfer, and then without chloride, it wasn't functioning. And another interesting aspect of the molecular dynamics simulations are shown up here, where what we see is that this is the position chloride is found in the crystal structure. But during the molecular dynamics simulations, chloride moved over to another position. And that suggested to us that maybe chloride is playing a dynamic role to modulate the pKa's of residues. So if it's close to an aspartic residue, it would make it less favorable to be ionized because of the negative electrostatic interaction. And maybe when it moves over here, it would be more favorable. So you could imagine gating pKa's to help facilitate proton transfer. So that was a hypothesis. So to test this, we decided we wanted to mutate this lysine residue to a neutral residue that had been found in other enzymes that require chloride to eliminate the binding of chloride. And so make a number of predictions. We hypothesized would eliminate chloride binding. It would also have effects on proton transport and some of the properties of the manganese cluster. I won't go into all the details, but just show some of the results on chloride binding. And in fact, so this was done with Rick Debus. He generated the mutant where he changed the lysine 317 residue to an alanine, and we probed its properties. And as was hypothesized, if you look at now the activity, oxygen involving activity as a function of chloride, you see that there's a chloride requirement for the wild type. We made an arginine mutant, and there's a chloride requirement, actually less tight binding of chloride, and so you see a more significant enhancement when chloride is there. That's largely because even at zero chloride, there's a fair amount already bound. But interestingly, when we mutated that lysine residue to an alanine, we saw no chloride requirement at all. In fact, chloride was somewhat inhibitory. So as you added high chloride concentrations, the activity was somewhat decreased. And so this was actually the first photosystem 2 that no longer needed chloride. It's not as good as the wild type. You can see that the activity is, optimum activity is maybe only about half as much as the wild type, but it works without chloride. So then is this really playing a role to facilitate proton transfer? So some of the properties of this mutant. So when we look at now flash oxygen measurements, what we're looking at is the period 4 oscillations of oxygen with single turnover flashes. And you can see in this mutant that the oscillations are less pronounced and they damp down more quickly. This can be modeled by a miss factor. There's about 22% misses in the mutant, much higher than in the wild type. And so is this due to protons? So we've been looking at this in some detail more recently. I'm not going to go into a lot of detail on that, but just to show one experiment that we've recently done that suggests that in fact this lesion is due to a problem with proton transport. And this is now using bicarbonate as a chemical rescue agent. So bicarbonate is a weak acid, and it can bind protons, and also it's diffusible. So we had hypothesized it might be able to carry protons past this lesion. And in fact, that's consistent with what we observed. So this is looking at now the miss factor in a flash oxygen sequence and comparing the misses in untreated, which is just no added bicarbonate, versus adding about 10 millimolar sodium bicarbonate. And there's no effect on that in the wild type, consistent with the fact that there's a intact proton transfer pathway. This is another mutation that is actually have a residue that's binding to chloride, and it doesn't have a big effect, and then doesn't also show significant misses improvement. But if you see the lysine to alanine mutation, we've got a high number of misses in the absence of bicarbonate, and with added bicarbonate, we recover the properties pretty much the same as in the wild type, which suggests really that this is functioning to help to facilitate proton transfer. So we're still looking in more detail on this, but I thought that was a nice story about how this project, which grew out of the Eastern Regional Photosynthesis Meeting, is continuing to ongoing. This is now the 36th Eastern Regional Photosynthesis Meeting was just held last month. And that there's a strong future for photosynthesis in the Northeast, and I believe also on the West Coast where Bob organized meetings. So thanks, Bob. It's been wonderful to have these meetings to create opportunities to collaborate. And let me just thank my group for the work they've done, is particularly the people who we collaborated with: Marilyn Gunner, Doug Bruce, and Victor Batista. And those collaborations have been facilitated tremendously by the Eastern Regional Photosynthesis Meeting. And thanks. I hope I'm about on time, and I'd be happy to answer any questions if I can.
Okay, so now we'll move on to the last graduate student and lab member talks. So this is going to be Bob's lab. We're going to give these quick 5-minute rundowns of what we do and our time with Bob. And yes, gazebos were involved. So that's an interesting thing about that.
Are you going to tell that one, or am I going to tell that one?
I think you're going to be the gazebo man.
Be the gazebo man. Okay, so
All right.
Derek and
Yeah, so we'll start with Ben.
so if Derek, Haijun, and Wenjin can also come down here, because we're all gonna. So all right, so. Oh, okay, I see. All right, so what we're gonna do is we're gonna have. I suppose we're gonna have each person come up as they do theirs, but. I thought that it'd be a really nice thing to have all of the final lab members in in Bob's lab come up and just give a little bit of a rundown of what they've been doing in their projects for basically the projects that will kind of wrap up Bob's lab as we close things down in the next few weeks or so. So anyways, I'm just going to go first. I'm the last graduate student in Bob's lab. Of course, we heard from Barry Bruce earlier, first graduate student. And also Raphael, Haijun, Wenjing, and Derek are also going to give a short little thing. So this is a classic picture you've probably all seen with Bob with his cyanobacteria. Now talking about oxygenic in this case. So I just want to thank Bob before we get started for providing a really great lab experience and some really fun research. It's been really great. And so yeah, thank you, Bob. And also thank you, PARC, because the Photosynthetic Antenna Research Center has been very useful in terms of collaborations, being able to go on these scientific exchanges and travel. I've gone to Australia to visit Min Chen, the University of Sydney. Went to visit Jerry Timlin at Sandia in New Mexico. So that's been really great, and of course a lot of people got to do these things, and it really fostered collaboration. Last year was our last all hands meeting, which is the picture here of everybody who was there. All hands meeting was always held over where the construction has been for the last couple of years, or next to, and so that's been really great as well. So today we're going to have talks from the 5 of us, and I'll let them. And this is, I like Derek's little sort of clown hat picture here with his cryostat, and I'm sure he'll talk a little bit about that. So, just a kind of a quick rundown of what I've been working on. So I'm also interested in being able to utilize more light into the far red as opposed to just the visible, but instead of by changing reaction centers, as Don mentioned before, instead we're looking to do this by looking at what nature has already figured out, basically in oxygenic phototrophs, particularly algae and cyanobacteria. So in a lot of ways, we find that chlorophyll F and chlorophyll D are some of the more common methods, but there are also a lot of eukaryotes. So that's one of my particular interests is how the eukaryotes do it. So actually, what I ended up doing when I first started in the lab was I went out and took some samples from the environment. These are just some pictures of a few of the different things that I found. This one here actually does have chlorophyll F. This is, these are all chlorophyll A containing algae. And so what we did was we used these custom-built far-red chambers to provide a selective condition using these little LEDs. I always have to have a picture of these in my slides. And they produce 740 nanometer light, and that provides a really strong selective condition for only things that can grow under far-red. And so one of my favorites was this isolate from Forest Park called the FP5. It was a eustigmatophyte alga. And it only contains chlorophyll A, but it's able to utilize light out to 710 nanometers, as opposed to the standard 680. And so that's always kind of interesting. This is just a light microscope picture of that. Some fluorescence confocal, and also we did some deep etch freeze fracture EM. So you can see that it's got a very nice absorption peak in the far red there. So that's, of course, why we're interested in it. And one thing I do, and just for some interesting images here, you can see that when we were able to grow this under far red light or under blue light, you have some pretty significant differences in the fluorescence. So you can see this is actually some of the confocal, hyperspectral confocal that I did when I went to Sandia. And so we can actually see that when we grow this under far red light, it adapts very nicely and actually builds this far red antenna that it doesn't seem to have under blue light to give sort of the opposite. So just some images there so you can see what makes up the merge. So that's going to be Raphael's. And I guess I've been told to do the gazebo story. So a couple of years ago, it was it was Raphael, me, and Jeremy was also involved, I believe. What? Yeah, he was. Okay. So Bob had this gazebo, and you'd gotten it, I think, like a couple of years before, and it was still in the box. So we got to go over to Bob's house on this really hot summer day, and we went and we set this thing up, and it was actually great. And it was all this big kit. It definitely required multiple people. I remember the beams getting nice and hot, so you couldn't really touch them without gloves at some point. Bob's got a solar system on his house. He captures some of that energy, but we were working in the sun. It was great, and we finally got this thing done. It's a beautiful piece. Bob tells me that it's still functional now, several years later, and that the side curtains have been removed. But other than that, it's still a pretty nice piece of equipment. So if you come over to his house at any point for the party tonight, you'll probably see the gazebo. I hope we get to hang out in it a little bit more.
All right, thank you. So for all of you who don't know me, my name is Raphael Serra. I'm Bob's postdoc. I came here about 5 years ago, and I've been tackling the FMO project. So FMO is a cool protein. If you don't know about it, read about it. It's really interesting. It's essentially an antenna protein that interfaces in the green sulfur bacteria, this large chlorosome antenna, with the reaction center. And many studies were conducted on this protein. One of the really interesting ones is the energy transfer studies, and what I find really interesting about this is the results that we get for energy transfer efficiency in our labs are always different from the European one. So I think the European C. tepidum might have something going on with it versus ours. I really wish that someone could do that experiment where they would just take the European strain, the American strain, and compare them under the same laser setup. I think that would be a very useful thing for the scientific community. But what I've studied is I try to look at what's going on the absorption spectrum. So it's very deceptively simple. There's a lot going on with this protein. There's about 8 pigments in each monomer. It forms a trimer of these monomers, and there's about 7 different excitons underlying the absorption spectrum of this species right here. And there's been many simulation and calculations done on it, but nobody's really been able to do any sort of mutation on it to actually biochemically analyze what's going on with these excitonic components. And so that's what I set out to do in Bob's lab. I created this expression system, which would then generate gentamicin-resistant mutants using a selection marker, and I would get these FMO mutants of and that was an interesting story because I spent a whole year doing this, and I get to do one slide, which is like a PowerPoint cartoon. So, and yes, Carl, I did use those chambers, those nice little cabinets with the lights, and that produced these FMO mutants. So I'm pretty happy that we had some light bulbs and wooden chambers. And with that, I generated a ton of mutants. I was looking at what the interactions were between the polypeptide and the pigments, and I was just changing the local environment to look at what the underlying excitons were. And I measured this with absorption shifts, both at room and low temperature. And we also collaborated with Sergei Savikin's lab to do some modeling on, just get a snapshot of essentially where these different bacterial chlorophylls are contributing to the different aspects of the absorption spectrum. So one of the cool things is that we actually biochemically confirmed that bacterial chlorophyll number 3 is that lower energy pigment, because when you make that mutant, all that absorption goes away over on the low energy side of the absorption spectrum. But also there's a lot of other features. So each one of these mutants produces very rich changes in absorption features that you can analyze. The other cool thing was that the 8th bacterial chlorophyll, when we changed it, it looked like it resulted in a loss of the pigment because it was hard to normalize the two spectra. So we modeled it as a loss, and we see a little blip here around 806 nanometers. So we were able to identify where the absorption of that 8th bacterial chlorophyll lies. And so that was a point of contention before, but now we think we've solved that, and I sort of solved it with several other mutants, but I haven't really published that. And another thing that I was really interested in is looking at the trimerization interface of FMO. So one of the burning questions was sort of, can we isolate a monomer? Because there's been a lot of molecular dynamic studies that have just taken, I'll just take an FMO monomer, throw it in a box of water, and simulate that without taking account of the whole trimer. And so I decided, okay, fine, I'll I'll hand these modelers a cookie and see if I can generate a species that would confirm any sort of results that they're having. And so I looked at sort of what are the interfacial important residues that are in the interfacial region between these FMO in this FMO trimer. And what I found out ended up being really interesting because I targeted these salt bridges, and I started taking these salt bridges away. I noticed two things. One is that you can't purify the protein. You can't get it out of the organism without it falling apart. So FMO turns into, it goes from being a rock essentially to a handful of sand. It just falls apart as soon as you take that protein out of its context. And you can't really isolate the species. It becomes very labile. And the other thing is that you form inclusion bodies in the organism, and they're much bigger than the chlorosomes. And I still have no idea what those inclusion bodies are. I thought they were FMO, but they're clearly not. So something else is accumulating in there. And that's sort of where I've ended off. And I'd like to thank Bob for taking me in as a postdoc. I had no idea really that he's been trying to get FMO mutagenesis to work, but I guess I sort of blindly attacked it. And I'm happy he was a very sort of hands-off kind of guy, let me do my own thing. There's always the horror stories about being micromanaged too much, so thanks, Bob.
Thank you. Here's the clicker. Okay, great. In case you don't know my name is Haijun. I'm a research scientist in Bob's lab. And Bob's for science is contagious, but he always expressed this in a very humble way. I remembered one time that when I talked with him that the photosynthesis research center is a really big thing, and from DOE supported the research center, and I told him that my experience is mostly on reaction center on the PS2 side. And he told me that a light harvesting complex is cheap. And then he switched on, you can bridge the cheap thing to your expensive reaction centers, which probably true in terms of biochemical preparation. It's very difficult to get reaction centers. Well, for light harvesting complex, probably it's much easier. So my research is mostly focusing on the interface of light harvesting complex and the reaction centers. What's the structural basis dictating the efficient energy transfer from light harvesting complex to the reaction centers, which is very important from many perspectives, from photosynthesis re-engineering and okay, so, the next slide. Okay, so as a biochemist, we tend to purify things out, but this will automatically disrupt the weak interactions of all the components, weakly interact with protein complexes. For example, PS2, biochemists try to get pure photosynthesis 2, but it turns out the activities goes down. And right now, we tend to have a scheme that for all of these pigment protein complexes, they tend to somehow weakly interact with each other. And, well, this still allows the efficient energy transfer from light harvesting complex feeding the hungry reaction centers, but also allows the regulatory components jumping in to fine-tune the excitation energy transfer and efficiency. And in this figure, you can see that there are quite a few data showing that actually for the light harvesting complex and the reaction center PS1 and PS2, they are distributed across the cytochrome membrane, but somehow they do have some patterns allowing light harvesting complex passing energy to both reaction centers. And you can see that sometimes the PS2 can be in a patch over here, and sometimes you can see this is PS2 as green, and PS1 may form these patches too. but also there are a lot of other areas that PS1 and PS2 and light harvesting complex, they patch together. Okay, so 2 years ago, there was a Nature paper published on the phycobilism. It's been a mystery for a long time. People want to know the light harvesting complex in cyanobacteria and red alga. And starting from here, I think my research is mostly interested in how the light phycobilism connect to the reaction centers. So based on the crystal structure, and we have all this either crystallized pigments or the in silicon predicted structure, and do the homology modeling into the core. And this is the whole process. And we actually swapped all of the cyanobacteria pigment components to the red alga system. It turns out there are a lot of interesting process coming out. And this also reminds me, 5 years ago, we have this science paper talking about the interactions of light harvesting complex to photosystem 2 and photosystem 1. And if we dock this light harvesting, the phycobilism into photosystem 2, constrained by our cross-linking data, and we can have this light harvesting complex in cyanobacteria just on the top of photosystem 2. And okay, so this is the top view of the complexes, and this is side view. And you can see this purple area is PS1, and in the center is PS2. And right over there is what we have observed, the cross-links connecting the light harvesting complex to photosystem 2, because that's what we thought, that the energy goes directly into reaction photosystem 2. Now here is the PS2 side view, and this top view from the cytoplasm, from the phycobilism side, and from there, and that's where the phycobilism feeding energy into the reaction centers. Now this is our model showing that how this phycobilism core touch into the photosystem 2, and all this red area are the crosslinks we found that connecting into the photosystem 2. And this is the bottom face that the crosslinks we found over there. This is measured by the Angstrom level, and they are connected to aligning this over there. That's the CP47 there, and CP43 over there, and this is a side view of a phycobilism core sitting on the top of photosystem 2. And not only that, and we also noticed that APCB, another component on the edge of over there, they cross-linked to the PSAA, the N-terminal loop area. So this is what we have reported in our paper, that talking about in cyanobacteria, there is some area that phycobilism feeding energy can go to directly photosystem 1 and photosystem 2, and this is this can allow the energy transfer, balancing or sorting out either one or two or both, or even photoprotection. So I go quicker. And another question we are interested in is trying to figure out what's the commonality between two oxidanic phototrophs, which is higher plants and cyanobacteria. And here is the top view of photosystem 2 super complex from higher plants. And this is cryo-EM data. And one thing I want to point out here is that over here, if you see this orange color, this we call this minor light harvesting complex CP29. It has a bridge, like a sticking out, holding photosystem 2 on the top over there. And it turns out that the energy of this light harvesting complex 2 can pass energy through this area into CP47. And if we look at the homology of this protein sequence with the cyanobacteria, the APCE, the loop area, they do share a low homology sequence. That's what we have observed, the area cross-linked to the cyanobacteria of photosym2. And show over there, that's the loop over there. And you may notice that the homology is not so significant. It's like a twilight zone, but still. So what's the structural basis about this? Will be very interesting to compare. And I'll pass this to. Oh, okay.
Where is the mouse? Oh. This is a problem. Go back one.
My name is Wenjing, and it's only 1 year since I came from China. So it's not a long time, but I do learn a lot during this period. It's helped me a lot. And today I will share you with 2 stories about the OCP-related photoprotection in cyanobacteria. Okay. OCP is really very beautiful protein, and the O here means orange. We can see the color is orange, and after highlight elimination, and the N-terminal and the C-terminal are separated and converted into red form OCP. It's active form. You can see the color here is red, and the absorption spectrum have a red shift. And the active form OCP, the N-terminal will bind to the fibroblastum. It's the light harvesting antenna in cyanobacteria. And this N-terminal bind to the core of fibroblastum and creates the harvested light energy as heat. And this whole process causes OCP-related non-photochemical quenching. And in darkness, there's another protein, FRP protein, will help OCPR depart from the core of fibroblastum and convert the red form OCP into an orange form OCP. And this process called fluorescence recovery. And the cyano we know always have several those to regulate one single process to make sure themselves are safe under different conditions. So we would want to ask: is there are some other factors involved in this process? So we found copper-1 can convert OCPO into OCPR and constantly induce the fibroblastum fluorescence quenching, even in the presence of FRP in dark. And based on the results we have, and we promote this model, and the secular copper-2 can reduced by light or reducing agents and into copper-1, and the copper-1 can bind to the OCPO protein and convert to the OCPR form, and all directly bind to the photoactivity OCPR, and we have this complex. And this can lock OCP in a red form, and can constantly bind to the core fibroblastum and quench, induce the fluorescence quenching. So. This model, this mechanism can protect cyano cells from photo damage and copper excess conditions. And my another story is about the binding sites of the OCPR to fibroblastum. It's still an open question, and here, it's the pink color is still RCP, and RCP only contains N-terminal and the pigment carotenoid. And this protein can bind to the core of fibroblastum and can quench the fibroblastum quenching as well as OCPR. So we compare these two protein structures, and we found very dramatic change difference. And in the OCPO, this area we show in blue color is a loop, and while in the RCP, there's shown in red color is the alpha helix. And among this area, we found we are very interested in leucine 51, which shows in blue color in OCPO and yellow color in RCP. And there's very interesting operation change in these two structures. We can see the yellow one is exposed and the blue one is exposed and the yellow one localized in the surface around the exposed ring of the pigments. And we can see here this the well-known arginine 155, which is suggested is directly involved in the binding to the fibroblasm, and so we think it's very important amino acid in the binding process. So we made the mutation and mutant leucine 51 to nonpolar adenine and polar polar and positive and negative charged amino acids, and then all the mutations, the absorption of the orange form and the red form are similar. And we detect the photoactivation, the rate of photoactivation were 2 faster times compared with the wild type, while the relaxing rates is similar, exactly the same actually. However, all these mutations cannot quench the fibroblasm. And interestingly, we found that when mutant leucine 51 into alanine and the binding activity to the fibroblasm is not changed. It's just the same as the wild type. While we change the amino acids into the we have these conclusions. So leucine 51 is distant from the carotenoid OCPO, but it plays an important role in stabilization of OCP, the orange form OCP. And the nonpolar radiate of leucine 51 is directly involved in the OCP binding to fibroblasm, but and played an essential role in the energy quenching process. So thank you.
Hello, hello. It's always be the last one before lunch, right? Okay, so I promise you it will be very short. So for those who don't see me on the agenda, I'm not on the agenda. And so initially I didn't want to give a talk because I first didn't want to give a research talk. First, I had I had a problem because I thought maybe I can find some pictures and just do like a history, because I've been here since 2009, and I started to put all these pictures from the laser facility I've been working in on the slide, and there's like me, is it pointer, me officially touring people, working alone in the lab, There's like nice picture of facility, nice pictures of laser beams, but there is one problem. There was one big problem. I couldn't find even one picture with Bob, okay? And me, okay? So how can you talk about Bob without Bob, right? And then, like a week ago, we had a short talk with Bob, and I got like inspiration from him, because he asked me, Derek, do you know how many papers we published together? And of course, I didn't know. And he said, I think 25. And like, oh yeah, this is good direction to go, okay? So, let me show you something, okay? It will be science from 10 years of our work in numbers, okay? Even better, in statistics, okay? And I know the last thing you would like to show to people is statistics, okay? So you have to sell it in the very specific way and to involve your audience also into it. So I thought that's a very good way to sell it, okay? And most people will know what I'm asking for, all right? So let's put this question one more time. If you cannot read it, how many papers we co-authored between 2009 and 2019? A, 10, B, 24, C, 35, D, 100. D, very optimistic. 35. So it's a really good number, I think, actually. And now think about it. You have 35 pages, not pages, you have 35 papers. In average, I calculated there's like 12 pages for one paper. So we have something like 400 pages of text, right? So now you have something what we call big data, you know? Now everybody wants big data from you, right? So what can we get from big data, okay? So I was thinking maybe we can run statistics and get like a very short summary what we did for these 10 years, okay? So here you have second chance, okay? What was the most frequent word we used in papers? So A, bacterial chlorophyll. B, the, okay? C, absorption, or D, spectra. All right, okay. Just one person, please. B. Yes. Like, no way, that's the whole summary of our work, right? So it's like, okay, you know, I said to my wife this result, and you know, she's like a linguistic person, like, come on, it's like the most frequent word in the world, okay. So like, dig more, okay, so, okay. I did it, okay. So, let's see. What next would you expect? Of. And these are real numbers, okay, that came out from it. We love off. We love end. 7,000 and more now. We love absorption. Oh, it's getting there. All right, what else? Fluorescence. Closer, okay? Photosynthetic. All right? Transient. That's something enigmatic, but it makes sense, actually. Spectroscopy. Okay, sounds very prestigious. Okay, and pigments. All right, and if you look at this, it's like completely random choice of, you know, words from it. But if you arrange it in the right way, that's all you get. Transient absorption and fluorescence spectroscopy of the photosynthetic pigments. Right. Ready for lunch? No questions.
Okay, so this is actually a bit of a tribute to Bob and Liz both, because one of their passions is outreach. And so what you're seeing here is a planetarium movie that has just been released. Let me tell you what this is projected in some strange circular manner. So this is actually meant to be projected in a planetarium, right? The bottom is in front of you, the center is on top of you, and the top of the screen is behind you. So you sit in the movie. And the subject matter is, well, it's been my privilege that Bob selected my research to be the cover of his book on photosynthesis. And so what you're seeing is the structural and functional modeling of the chromatophore from purple bacteria. And this is about 15 manuscripts over the past decade, all of which are collaborations with Neil Hunter, turned into a story for 9-year-olds. And we worked with Donna Cox's team at the NCSA in Urbana-Champaign, who is, of course, a world expert in turning science into animation. So this is far beyond my ability to create by myself. And we've worked with the writer Tom Lucas, because to make something understandable is as hard as doing the research in the first place. And the narration for the movie is actually done by Richard Dormer from Game of Thrones. So this just got released a few weeks ago. The world premiere was in Germany about a month ago. And we just learned that we have received an official selection from SIGGRAPH 2019, which is a computer graphics conference. Just to give you an idea, the previous year's awards included Pixar and Neil Blomkamp. So normally scientists don't make the cut. It's a bit of a rare honor for us. So just to give you an idea of what the backdrop is, so there's an extensive collaborative network, both for the animation side and for the computational infrastructure, which was, of course, facilitated by Klaus Schulten's legacy, and the experimental collaborations at the center of which was Neil Hunter. And this was grounded at PARC for the last several years. And so I can't take time to actually show you research, but catch me offline and ask me about what we are doing for the granule and cyanobacterial thylakoid membranes, which I hope will be the next iteration of such a movie. So on that note, thank you, and thank you, Bob, thank you, Liz.
Hi, everybody. My name is Greg Orff, and I was a PhD student with Bob from 2010 to 2015. And when I was in Bob's group, I worked primarily on green sulfur bacteria, specifically chlorosomes and FMO. You saw a little bit of what I did when Govinji gave his talk a little bit earlier. But one of the things that really stuck with me from my time when I was in Bob's group was just the genuine excitement that Bob showed whenever I would come into his office with some new result or something interesting. If I felt compelled to do an experiment, for some reason, if it wasn't necessarily one of the aims on our grants, and I still felt compelled to do the experiment, he genuinely showed excitement. And you know, that's some of the best work that I published was actually that came from experiments that I felt compelled to do that weren't necessarily on my to-do list. So. You know, here's just a few pictures from my time. The first picture here on the left is the group from 2014, and if I'm not mistaken, it might have been the group at its largest point. I think at one point we had 20 people, including undergrads. So I have to be fact-checked on that, but it felt like I was constantly running into people, literally in the laboratory. But we were extremely productive, which was great. And in the middle here is when I graduated in 2015. I always thought the hooding ceremony was a really cool way to end your PhD. And sort of a funny story here on the right, this was the Western Photosynthesis Conference a couple of years ago, and I won an award for a poster. And it's not every day that your PhD advisor autographs his own textbook to give you as a prize. So we toss the word humble around here with Bob, but you know, keep that in mind. But anyway, the last point here. You know, I left Bob's lab 3 years ago to take on a postdoc at ASU, and pretty much every year since I've left, some of the work that I've done is still being published. And what that comes down to is the fact that we had such a creative and productive environment in Bob's lab, and he really gave us that environment to explore the questions that we were trying to answer in the fullest extent possible. And here's just kind of a picture of sort of the last bit of work, at least I think it's the last bit of work that I did that's going to be published soon. It's a neutron crystal structure of FMO, which I think is going to be really exciting for the people who do computational analysis of photosynthesis, especially energy transfer. So with that, again, Bob, it was great to be back and visit everybody and see you. Hand off to the next speaker. Thanks.
I'm George King. I worked for Bob 1986 and 1987, finished my master's degree in 1991. I was looking through my files. I did not find any pictures of the Bob or the lab. I did find an article from the 2005 in my folder marked Dr. Blankenship. a deep ocean photosynthesis found article, June 2005. I was looking to hear Bob's words from the past. Blankenship is in New Zealand and could not be reached. So we did not. Okay. Let's see. When I was there, I don't remember a light bulb in a drawer that opened or closed. I remember an actual thing with a bunch of lights around it, and that was low light conditions, which it seemed like it'd be higher light conditions. I have a shirt, the early events in photosynthesis. I have long outgrown it, but I do treasure it. John reminded me, John Freeman, that I did a bunch of computer stuff to run the equipment that took the photosynthetic data and plotted it when you actually had to have a program to plot things. It's insane. And I tried to make it professor-proof, and if you push the wrong button, it would make a sad sound. I was told I don't remember this, but I was told within 5 minutes Bob pushed the wrong button, and I didn't catch it somehow. And anyway, you're going to hear what a great guy, I call him Dr. Bob, because that's what we call him in the house. You all are PhDs, I'm not. And I will confirm he's supportive, gave direction, and wonderful. And we disagreed often, but very pleasantly on politics. His autographed copy of his book sits right next to my autographed copy of Milton Friedman's Free to Choose. I treasure them both. Let's see. A lot of you are going to look back, and like the last speaker and almost everybody else, how much of your professional life has evolved from where you were with Bob. I went directly from Bob's lab to law school. So, but I don't, for a minute, think that was, time wasted or anything. I thought it was wonderful. And after the second semester of quantum mechanics, law school wasn't that bad. Let's see. In fact, I had a court appearance scheduled as a telephone for this morning, and I had to file a motion to say that I had to be here instead of do that. And so I stapled on like the 3 papers, not 35-3, that I had done with Bob. And then I got an email from my very smart client who said, in terms of that it was sent to opposing counsel in the court, you're just showing off, aren't you? Yes, I was. I was showing off that I got to work with Bob. My chemistry has been fun during Breaking Bad. Everybody at the office wanted to talk about making meth with me. Bob came to my wedding and told my wife, make sure that George finishes his master's. I hadn't gotten it done quite yet. And so but I did the next year. But I was working in a law firm, and I'd send drafts of my thesis to professors. And finally, somebody said, stop sending us big envelopes from law firms, please, stop. So anyway, oh my gosh, I have 40 seconds left. I got through all that. I've really enjoyed staying in touch with Bob and Liz, and I've heard Liz's name 1,000 times. I don't know if we've met. I think we have maybe once. But I really like it, and I'm just really glad to see you all. And I really thank everybody who took all the time and effort to put this together, because I come at it from the perspective of my life's research and all that stuff is, like most of you, unlike most of you, doesn't look back to Bob, but who I am as a person and how much I enjoy looking at the world through as a chemist and a researcher, even just for a few years. I really appreciate it. And my time's up.
Hello. All right. Hi, everyone. Aaron Collins. I was in Bob's lab from 2005 to 2010. And we were having lunch today with some of the folks. So I started at Arizona State University with Bob for about 1 year before he got up and moved to WashU. And I was very happy that he allowed me to come along for the journey. And while we were at lunch, sort of reminiscing about my time at ASU, the story of the couch came up, Bob. So I don't have a slide about this, but so for those who were in the lab, Bob quite frequently would mosey on down to the lab and sit on this sort of tan couch and just see what was going on in the lab. I think it was a way to escape from maybe department chair duties every once in a while. But it was always great to see Bob pop down. We'd pour thick black pots of coffee, enjoy it, and talk about what we were up to. And when Bob invited me to come to WashU and continue my PhD journey, he also allowed me to help with the lab design. So when we were building the lab design for lab sciences, we were thinking about, all right, where do we put holes to run FPLC lines and things like that? But the one space we absolutely had to have was for that specific couch. So we moved this couch from Arizona. It's just a $50 IKEA couch maybe. But we couldn't get rid of it, right? And that couch, is it still there? It was still there? Oh, we should have put it like in a museum. No, I know. Oh. Oh, terrific. We should go visit it. Well, anyway, we have a lot of great memories of just looking at spectra on that couch. You know, you can tell a lot of information of your research just looking at simple spectra. But I want to point to some really nice memories I had from 2008. So every year, Bob would have a holiday party. I think every year I went to Bob's holiday party. And this is for some really great people in 2008. We had Jiro Harada, who was visiting our lab for around 6 months around this time, and I got to know him really well. In the bottom corners, you'll see a lot of hilarity going on. And that's because for many people in this photo, it was their first time doing a white elephant Christmas exchange. So for those who aren't familiar, this is where you have some functional but gaudy gift, and you sort of trade them around. And so we had things like this giant green Hulk glove, gaudy lamps. It was a really great time. And I don't know if that tradition continued, but it's one of the memories I have from this time that I really enjoyed. But yeah, so you'll notice everyone smiling in these photos, and that's just because working in Bob's lab was always much more fun than it was work, so it seemed. Especially my experiences since then. It was probably the most fun I've ever had doing a job you get paid for. So anyway, I'll stop there and say thanks, Bob.
Okay, my name is Hong Chen. I work for DOE, and I'm really glad that Bob got a lot of funding from DOE and make a great contribution to science. My first association with Bob was in 1990 when both my wife and I received a research scholarship to study under the Center of the Early Event in Photosynthesis. George just showed the T-shirt that he had. So that's where we come to SU to study. And for me as an international student, and it's really hard for me at times because Bob probably will remember at times my English is really not that good at all. And Bob was very nice and kind and very approachable, and so helped me go step by step and to overcome each challenges. And I really appreciate that. And two of you show a lot of a holiday party, and I want to show you this picture here. Bob probably remember this is the flyer he sent out for the Halloween party at his new house in Canyon Drive, if you remember. So we found the flyer before we came out here, and so we thought that's really good. And Dr. Govinji and shared some of the early photos of that, similar to that. So at time he said, he had changed in the first 25 years, and that's in 1995, and he was looking like the lower picture here, okay? And so that really is a huge change compared to him now and 95. There really not much change at all. So yeah. So, but anyway, my memory to Bob is, if you look at the photo right there, he's, although he is very productive in research, and he is also very humorous, and just look at picture here. And from picture here, you can also see he smiles a lot, and he has a lot of energy. We all experience that. And he has the ability to bring out the best out of the people working with him and around him. So I really appreciate that. Okay, I also want to bring you back into history. In 1991, in one afternoon, in October, okay, earlier in the day, there are some undergraduates working in the lab. Of course, they are using solvent and so forth and so on, and they put all the solvent all together into the hood and into a jar. So what happened would be, if we put nitric acid into mixed with organic compound and all that, compounding that, the undergraduate screw up, tighten the lid. So it become what happened. So we have explosion. So I was there, and so was the cleaning lady. And Bob's office was right next to the lab, and he was fast to respond. And he put me under the emergency shower. And emergency shower does work, and the water is cold, I can tell you that, okay? So we have a hazmat response and all that, and I got my first ambulance ride to the hospital. I hope that's the only one that, in my life. But thankfully, I was okay. And after that, we all have a good lesson learned, and we put on the safety cap and has a heightened awareness. And I'm sure after that, there's nothing like that happen in Bob's lab. So that's great jobs. So, but again, looking up Bob's publication right here at Liz Farewell University, and do you know how many percentage of the paper he published as the total here? It's more than 50% of his publication here. So I'm really glad he has a great time and a great career here. I would say thank you very much, Bob, for your help.
Okay, so I'm Erica Wunderlich Majumdar, and I was graduated in 2015, so one of the more recent ones out of the lab, and this is just kind of a myriad of photos I was finding from the time we were working. And just one note on the couch, we were explained that it was for our overnight samplings when you had 18-hour kinetic experiments, not for chatting and pots of coffee, but things change. Yeah, so up there, we have our classic sucrose density gradients. I think I was the person that killed the Rosiaflexus and the Chloroflexus project for the lab. Greg was saying his papers are still coming out. And those ended with me, and then also the Winogradsky columns. You can't have a Bob without a Winogradsky column, I felt like. And working with PARC, we had a lot of outreach opportunities, and so that's a bunch of us sort of being goofy, and Greg had the same picture of the lab at the same time, and we heard about the Eastern Photosynthesis Congress and the Western Photosynthesis Congress, and of course the Midwest and Turkey run. We had a lot of great memories hiking and sometimes getting lost. And then when we hosted the International Photosynthesis meeting here in St. Louis, that was very fun. I remembered Bob telling us we were going to get to go to an international conference, and then 3 years later he told us, guess what, I'm hosting. And so that we got to stay home for that, but it was really great. And then, of course, my defense a few years ago, and Bob has a lot of wisdom in a lot of different ways, but sometimes I think Bob didn't quite know what to do with me, to be honest. I think I was a little bit different, but as others have said, he was really hands-off and let us go. And so he would often tell me things like, I really wonder what you could do if you spent more time thinking about your science. You don't remember telling me that? I mean, I still publish plenty. We got 5 or 6 papers out, but I think I was involved in a lot of things, and so he often that. And he'd always also tell me that his job was to straighten my path when it was getting a little too windy, but not to set it for me. And that's certainly something I've carried forward with the other students I've worked with as I've gone on. And actually, I started my postdoc with Judy Wall at the University of Missouri, and she did her postdoc with Howard Guest, and so everything was sort of triangulating for me today. There's a lot of good photosynthesis history in that world, and in my training, and now I'm out at Scripps doing mass spectrometry, and so I should have paid a little more attention in Dr. Gross's class when I took that. And yeah, Bob also told me that I had this golden touch with fellowship writing. He would always say that too, so I thought that was funny. It was very true. I have no idea where I am on my time, but it was so many fond memories, and really instilled this passion in me. When I started here for chemistry, I thought I was going to do inorganic synthesis, and then I saw Bob's poster, and I joked that I traded up for a bigger ligand system, and and I haven't really looked back. I still look at electron transfer around metals. I just went further down the redox gradient, and still look at anaerobes and all of that. And so it's really set me off on a good path. And so thank you.
So I had to put some slides together so I could stay on time because everybody who knows me knows I have a really hard time giving a short talk with few words. So that's what I did, some slides for this. So I didn't want to overcome, George, I've got the same shirt that George has, except mine fits still. And so, which I'm happy for, but it was a great time at ASU. I'm Jeff Trost. I was Bob's first lab technician at Arizona State University starting back in 1985. And so it was a real flashpoint for my wife. At the time, it wasn't my wife, it was my roommate. At least that's what we told her sister when we moved out together. And so Bob was, Bob was a real flashpoint. My wife's an RN, and she'd passed her boards, and so she was going to get a job easily, and my job was going to be hard because I was just a lab rat. I was fortunate as an undergrad, and I'm glad Bob has been supporting so many undergrads. I was fortunate as an undergrad to get to do some real good work, and when I went to look for a job, Al Bieber says, oh, there's this new guy, Bob Blankenship, downstairs. I think he's got a job because he had filled his job. And I walked downstairs, and Bob had some time, and we ended up having a great conversation. My poor roommate at the time, now my wife of 33 years, she was sitting out on the bench outside with her fingers crossed and her legs crossed and everything going, oh, I hope he gets a job. I hope I don't have to take care of him all the time. So I actually got the job because serendipity, when I say serendipity and favors those prepared, when I was an undergrad, I did a lot of stuff you guys are still doing. I did a lot of protein cross-linking. I've done a lot of protein purification, and I did a lot of STS page gels, and those were exactly what Bob was looking for, so it actually is very serendipitous to find Bob. And it's, like I said, it's been a flashpoint. We talk about people with flashpoints in their lives. Well, getting to work with Bob was a flashpoint for me and for my wife as well. So I wanted to say kind of, I think Bob is a scientist who manages people. I'm more of a manager now who understands science. I don't do as much science as I used to, but I did understand everybody today pretty well. But what I think about in management stuff is saying, what is a good manager? And I started thinking, what is a good mentor? What is a good thesis mentor? And you say, I crossed out parent up there because they're not your parents. Your parents are your launch you. And I think it's just in line with what you just said, that thesis mentors guide you and they straighten your path and they give you a boost to go forward. And that's all the stuff that Bob did for us. Since I kind of thought I took some martial arts, and sorry, Bob, I know it's never like that. But in any case, I think that's what a good sense. So then I thought about a bunch of words saying, what would I think about Bob in our time there? And the one that comes in this forefront to me is integrity. We had a couple of people in our laboratory who were doing some things that were not really what they were supposed to do. And actually, Bob took care of it. He was concerned about it, and he did, we had one guy who was kind of a pathogenic liar, and Bob had to go talk to him about his problem and take care of it, and he was really concerned about it. It was actually good he showed a consternation about it, but he also took care of the job, and it was always that you had to have the right integrity. Having integrity has served me well for all of my career. It's something that carries forward, and Bob did that for us. And also curiosity and honesty. And I say cyclist, and cyclist we say is kind of a joke, because back then we all rode our bicycles to ASU. Bob would ride in the morning, he goes, I'd have a great idea while I was riding my bike on the way in the office, okay? And everybody in the lab scurried away. They were great ideas, they just weren't very pragmatic unless you wanted to spend 10 years in graduate school. So they were great ideas, but it's like kind of the more blood in the legs, not enough in the head. And they said, have a cup of coffee, sit down on the couch. We had a couch too, but we all did a lot of cycling. But again, all the acumen, and it didn't come up as high as I wanted as Bob's social acumen. A lot of scientists aren't great social people, and Bob actually is a hugely social person, and how many people he knows and how many friends he has and how he's made friends with everybody. It's something all scientists need to take as an example of how you can be a good social person, have good social acumen as well. So you can have this slide and try and think what you think. See if the words are right, Bob, okay? So, and then I wanted to just point, my last thing here was, this was my graduate gift from Bob, and he didn't sign this one, okay? This was back in, give me 1 minute. This was back in 1990 when we had a small Mac computer and spell check didn't work really well. I'm a terrible speller, still a terrible speller. So when I graduated, I got my thesis, and this is the 1988 version. It's still on my desk now of Webster's Dictionary, which I still have. But that's not the only thing I got from Bob. It's like all of you, we talk about all the great sincerity things, all the good work he's done for us, how he's made our best life for us in the laboratory. So I want to say, Bob, Liz, you can and you should be proud of everything you've done for all of us and how many people you've taken forward to enrich their lives to make great paths for it. So thank you for all you've done, and thank you all. Sorry I'm over. I can't find my drink ticket.
Thanks. Let's see. I'm Allison. For those who of you who don't recognize me, that's okay. I was actually never part of Bob's lab. I'm more of a recent, I guess, member of Bob's huge circle. Right, I work out at Stanford with WE and one of my fellow colleagues, Pete. And together with Bob and Nikki and Hydrogen, we recently have been working on the orange carotenoid protein, which I heard a couple people talk about earlier. And so they were kind enough to send us a bunch of wonderful samples for us to test. So another confession, I'm not even a biologist. I'm, I guess, more of a biology enthusiast. And so we were checking out the orange carotenoid protein in our single molecule traps and doing fluorescence spectroscopy. And the cool thing here was you guys use these, at least from my perspective, really elegant and intimidatingly complicated biochemical techniques to investigate the details of these systems, when what we do is we just kind of throw it in solution and then watch the fluorescence and see how it looks. And so we did that with the orange carotenoid protein on the phycobilosome, and we trapped it. We do the single molecule trapping technique where we can watch how it fluoresces over a long period of time. And some pretty cool results came out of it that we were excited about. So just by looking at the thing, we were able to see that there are actually two quenched states. That is, they're dim and they have very short lifetimes. So this is something we published recently, and it was a lot of fun to talk to Bob and to Nikki and to Hydrogen about what might be going on in the system and trying to explain why we were seeing two states instead of just one, which is what we'd expected. And I'm really grateful to be here and to meet all of Bob's wonderful alums. You guys have such a great community. So thanks for including me today, and thanks so much to Bob. Congratulations.
Okay, this is my solution to the requirement of just one slide. It's crammed with a lot of subsections like some figures and papers. And so one thing about working with Bob, he always had interesting research ideas. And so when I was collaborating with Bob about 32 years ago, the idea was to investigate the chlorosomes of the green gliding bacteria, or filamentous bacteria, Chloroflexus aurantiacus. And so the green color comes from this pigment bacterial chlorophyll C, which is up here. And it's very similar to chlorophyll A in terms of its absorption spectrum. And structurally, of course, it's similar as well. So it has one of the pyrrole rings is reduced in exactly the same place as on chlorophyll. There are some significant differences. So where chlorophyll A has a vinyl group, this has a hydroxyethyl group. And in chlorophyll A, there's a methylated carboxyl group down here. It's replaced by a proton. And what this allows is you can have pi stacking when molecules overlap, and I'll come back to why that's significant. Well, you can see up there kind of why it's important. But the idea of, and so what bacterial chlorophyll C does is it exists in chlorosomes, which are shown up here, and in Chloroflexus aurantiacus, the chlorosomes are ellipsoidal structures that are about 100 nanometers long, 30 nanometers wide, and 10 nanometers thick, and they're adjacent to a photosynthetic membrane that has the bacterial chlorophyll A containing reaction centers. Now, bacterial chlorophyll C in solution has a spectrum, as I said, very similar to chlorophyll A. In fact, you can kind of see it over here. It's a red peak around 660 to 670 and a Sorey peak around 430. But in chlorosomes, it shifts to 740, kind of like shown here. Although what's shown there is actually not a chlorosome spectrum. I'll come back to what it is a little later. But each chlorosome has about 5 to 10,000 bacterial chlorophyll Cs in it. And the purpose of those bacterial chlorophyll Cs is to absorb light and then transfer the excitation into the underlying membrane, as I said. And so what Bob wanted me to do is to work on what would be a reasonable structure for that antenna in the chlorosomes. And there were a couple of ideas. One is would be a bacterial chlorophyll C protein complex. But Alexander Krasnovsky in Moscow and also Kevin Smith in University of California at Davis showed that bacterial chlorophyll C in a non-polar organic solvent like hexane would aggregate, and it would give a spectrum very similar to the chlorosome spectrum. And so that's what this is. This is an aggregate that has peaks shift. So this peak is shifted to 750 nanometers, and the Sorey peak shifts to about 460. And that was formed by just taking a concentrated solution of bacterial chlorophyll C and methylene chloride and adding it to hexane. Now, I was able to get that aggregate purified a little bit more by noticing that if you had a concentrated solution of it in a cuvette, you poured out the contents of the cuvette, actually a lot of that aggregate had precipitated on the inner faces of the cuvette. And so filling it with fresh hexane gave a spectrum that was this solid curve down here. Now the dotted curve is a spectrum that was taken by putting a dried film of purified chlorosomes and drying it on the interface of a spectrophotometer cuvette, filling the cuvette with hexane, decanting the hexane. And what that did is that had no effect on the bacterial chlorophyll C absorption spectra, but it removed the carotenoids, which chloroflexus had a huge number of. In fact, the Aurantiacus means, even though it's called a green filamentous bacteria, it's sort of a golden color because of all the carotenoids. And so you remove those carotenoids, and what you see is a spectrum that matches pretty much perfectly the spectrum of the bacterial chlorophyll C. The differences are there's a peak at around 795 and 800, and then some differences in the Sorey region. Well, those would be characteristic of the bacterial chlorophyll A that's in the base plate that mediates transfer out of the chlorosome. And so that was a pretty good indication that what's really going on is that it's forming an aggregate inside the chlorosomes. And so then, of course, what we had to consider next is what's the structure of that aggregate. And so considering the properties of, certain properties of the bacterial chlorophyll C are required in order to have aggregation occur. It has to have the magnesium in the center of the molecule. If you convert it to a pheophytin, you don't get the aggregate. And it has to have the hydroxyethyl group. If you make pyrochlorophyll A, which is almost identical structure, I mean, some differences. Bacterial chlorophyll, or chlorophyll A doesn't have a methyl group here, and the esterifying alcohol in chlorophyll A is phytol, and in bacterial chlorophyll C, in chloroflexus, it's sterol, or octadecanol is another name. But anyway, it has to have the hydroxyethyl group. And also, we did some FTIR measurements, and as had Mark Lutz done in France, that showed when the aggregation occurred, there was a strong shift in the vibrational spectrum frequency of this keto-carbonyl group here. And so knowing that all those things were necessary, this may have been Bob's original idea, but we sort of came up with this idea of a structure for the aggregate. And what you have is the magnesium. On one side, it's ligated by the hydroxyl group of the hydroxyethyl, and on the other side by the keto-carbonyl. And that seemed like a pretty good idea, because if you line up, so the excitation for the long wavelength band is polarized along this axis, and in this kind of arrangement, you line up those axes, or those transition dipoles, parallel to one another and along the length of a oligomer that forms, and that could account for the increased intensity of the red band of the pigment complex and also the increased absorption. And so that looked pretty good, but the problem was that people had done some resonance Raman work, and they said that bacterial chlorophyll C all right, this will be very quick. So could not be 6 coordinate, it had to be 5 coordinates. So then these other models were suggested, and which you have, this is just dimers linked together by hydrogen bonding to the keto-carbonyl and the hydroxyethyl that ligates magnesium. And the other one is a similar kind of thing. That first bacterial chlorophyll ligates magnesium of the second with its oxygen and forms a hydrogen bond to the carbonyl of the other. And so this would, and shows the planes of the pigments as tilted. Doesn't necessarily have to be that way. I think actually newer models are a little bit more like this, so that you can have these chains right next to one another, so that you'll have something like this, but next to it another one with the oxygen forming a hydrogen bond here and no ligation of the magnesium and so on. And so now you get a two-dimensional structure that could even wrap up and be three-dimensional and make sort of a cylinder or something like that. And so the chlorosome does have rods. That may be a possibility. And finally, there's a picture with Bob and Liz in it. And this is, Bob was very popular with all of his colleagues at Arizona State University and frequently is there during the Christmas, New Year's visits, and he's always well received by the colleagues. And so this was a party, and probably a lot of you recognize Heather, who helped Bob move from ASU to here. And then one final little story about Bob. He was also very creative in dealing with the hot weather of Arizona. I remember that he would get his T-shirt soaking wet with water before he headed out the chemistry building on his bicycle to go home, and that would keep him cool during the ride home, and by the time he got home, his T-shirt would be perfectly dry again. And so he knew how to deal with the Arizona weather. And that also, Bob's informal dress style, I think, correlated well with his general informal personality in the lab, which always made it a pleasure to work with Bob.
Hi, I'm Melissa Del Rosario. I have notes so that I won't forget. I was a grad student of Bob 2001 to 2005. Okay, thank you. Okay, so unbeknownst to Bob, I had a blog. But before that, I was in 2000, I was in the Philippines applying to grad school in Arizona State. I got accepted, but no lab. They sent me a list of professors that had opening. I think I went alphabetical, so B was pretty high. But I saw I was interesting research, so I joined. But the first time I met Bob was him picking me up at the Phoenix airport, dropped me off at the lab, and that was my 5 years in Arizona State. All right, so it was my window to life in general in US, and I had so much fun, actually maybe too much fun, but I had a few, this was before Facebook. Remember, it was 2004, the word of the year was blog, you know, it was really, every entry is 2 days before I can upload something, but then over the weekend, I was reading it. It's kind of depressing a little bit, but mostly fun memories. I ended up reading my whole blog over the weekend. Lots of memories of repairing the ultracentrifuge I break in the middle of the night. And I had to change the vacuum pump oil in the janitorial services. It's like, who are you? Why are you fixing the vacuum pump underneath? And it was, you know, because it was a chemistry department, it wasn't yours, right? We just shared it. And so that was currently the blog is offline, it's not safe for work, but I did use code names before just to be safe, and Bob was called the boss, so yeah. So this was one of his pictures. I always thought that was kind of his personality, very relaxed, Hawaiian shirt. One of the things that we do in the lab is around 11:30 to 11:45, he comes to the lab to look for people to eat lunch with and to walk over to the Memorial Union, which is great if you have results to talk about, but you don't have it every day. So I kind of like, around 11:30, everyone disappears. And it's like, and we actually, or I suddenly have to run a gel or something. So he comes back, but found out that 3 topics are very neutral, you can talk about without having to talk about your research. So 3 topics, safe with Bob, which is a life pro tip for me because I learned that it applies to everywhere. One is weather. Arizona was like, oh, so hot. Yeah. Second is drinking, like he had a lot of drinking stories, and also what kind of beer is good. And the third is sports. You know, baseball, and I'm sure that's why he moved to St. Louis, probably. But yeah, I moved to the U.S. I didn't know what baseball, and I had to learn what the pickle is. And now, even football, I can call an illegal forward pass, and so I do that. So, but he also had a story that nobody believed when did you really chew parafilm as a chewing gum in the lab? Like in the olden days, I didn't have nobody believed me on that one. But the blog was the best of times and the worst of times. And this is after 4 years of growing chloroflexus. Finally, I had a tube of blue protein, and everyone was envious because blue protein it's not light sensitive, it's room temperature. So everyone hated me because everyone who had to work in the dark, so that because it's photosynthesis. But I had the blue protein, and I was so happy I took a picture of it, and that was the last of it, because the next thing I knew, I forgot what happened, but there was some EPR spectrum ready, and I had liquid nitrogen. I think I did not cool down the glass, so it broke, and so the next thing I knew, it was on the bench. I had the bench paper, and I had the blue stain on it, and I kept it there for a while, and I was like, I'm never gonna graduate here. But towards the end, Bob let me finish, and so I did recover eventually, and so the day I put it on my blog, like I erased that, and so I put PhD. But thank you, Bob, for writing all the recommendation letters. I think I had to ask you many, many years after still, but it was one of the greatest times in my life is those years in Bob's life.
Hi, I'm Pei-Ling Chen. I graduated, believe it or not, 26 years ago. And it's great, we're still doing fantastic research, but I want to share with you some of the characteristics and wisdom Bob had for me, for the long-lasting impact for my career and life after I graduate. So we all realize that Bob is a very successful master collaborator. I think I tried to reflect over the years, and when I left school, I was not able to, realize so many things. But now, I just wanted to share with you some of the very simple stories, which I think can show Bob's wisdom and his character. One time, Bob always tried to keep things simple. When I got back my first paper draft, my first author paper, there's a lot of red inks, on the paper. So Bob told me, if it's your experiment, if you are the only one who really knows this, nobody else in the world is doing this, that's why we're writing a paper. And you need to write a paper as if you're talking to a fourth grader. If a fourth grader can understand, you know what you're talking about. And this is so true. Until today, not only I have a habit of writing very simple messages, straightforward, and also I've been telling, I have my own company now, so I always tell people, if you don't know what you're talking about, don't even write it. And then in business, many times people try to, show how great they are, or they try to brag about things. You just ask them questions as if you are fourth graders. And if they can explain to you really well, and they know what they are talking about, otherwise, just leave them alone. So this is a very good, and also Bob has the unique way to instill confidence, in people. Like when we were 22 years old. So I was a lot of times trying to make the laser lathe down in the basement, and sometimes I get very frustrated. And then one time Bob came to me saying, you're going to get this work. Even I can do it. So I was thinking, he thinks, I can do it, meaning so he has his own ways to, give you confidence. And another time, I wanted to be exposed to the bigger community, so I asked to go to Japan to make a presentation. And I think he was not there, but we had to rehearse the talk. And then I was a bit timid, my voice kind of dies down, when I was giving a talk, doing rehearsal. And then he always tells me, hey, Pei-Ling, you are the only one who did the experiment. You know your substance, be confident. So that's another example, very small examples, but that carries, in many years in me, and I always tell my people the same thing. If you do your thing, do your job, you are the only one who know it, so be confident. And another area, I think I learned this in the hard way, because I think I'm the first fresh off the boat student Bob had, international student. So I thought, this is like the standard deal, as for a boss, a mentor. I feel like I can always, come to him, go to him and tell him about my ideas, or disagree, or complain even, say, how come I cannot go to Gordon conference, things like that. But boy, did I learn, a hard lesson later on. So I was just telling Bob during lunchtime, I had, many good careers, but I couldn't hold on to a job for more than 18 months for whatever reason. I want to learn more, or the boss is wrong, or so I ended up starting my own company. And one last thing I want to share with you is one time during my third job, this is 20 years ago at Merck, for some reason I got hold of my personal file. And I did get to see Bob's recommendation, reference letter for me. So this is a really weird feeling. Usually, you go to a next job and, you look, forward to the next career. So after I read the letter, I was thinking, I wish I could done one more experiment and did one more paper for Bob. So. But thank you for this opportunity. We didn't get connect for many years, but this is a great opportunity for me to express my gratitude to Bob, Liz. Thanks.
Take the mic. I'm Bob's first PhD student at Arizona State, his second graduate student, I guess. And I have 3 short stories and one that I already got taken away from me. I do also have a T-shirt. It is now in the rag bin, and I use it to clean the chain on my bicycle. So it's not in any condition to wear. But I also had to avoid Bob after he wrote in because he would read a paper for breakfast. And write in and come in with an idea that was going to mean you had an extra month's worth of work to do. I particularly avoided him during the last 6 months of my tenure because my wife had already moved on to her graduate career at Pennsylvania State University, and I was sort of trying to catch up. And one of the things that happened was he wrote in, and I was usually the first one in the lab, and he was usually the second. And I was goofing off. And I believe Erica had the straightening out the winding path. And Bob walks in and looks at me, goofing off. And he says, "I'm pretty certain that you have something better to do." And this was high criticism from Bob. This was the cruelest thing I had ever said to me. And I told, and my wife wishes this was the cruelest thing that had ever been told to her during her graduate career. But I actually remember that incident, obviously, and I actually kind of sit down often and say, is this the best thing I can be doing? And while I don't always give an honest answer to that, most of the time I do. And it's been a great thing to work by. And I didn't realize there were other people that didn't work on green proteins in Bob's lab. I do know that Jim McManus didn't, but I worked on cytochrome C554. Dr. Bruce, I can't see you. Thank you for leaving that for me. So I was, again, one of those people that got to work in the light. It was kind of nice. It wasn't oxygen sensitive. It was great. And so the other thing is I'm probably one of the few people with a PhD in chemistry that has placed a 5-bonded carbon atom in their thesis. And it's the first slide in your thesis presentation. And the organic chemist looks at you and says, "Really?" And you have to look at the organic chemist and say, "It's wrong." And that's the second thing I want to share is that sometimes that's the right thing to say when confronted with evidence that is incontrovertible about what you've done. It's been a pleasure to have been your first student, and it's been an honor to have been your first student. Thank you very much.
Hi, Diane Lentz. I graduated from Arizona State 18 years ago from Bob's Lab, 1995 to 2001. And here we are, fabulous, fun day of my life. And being here and seeing all of our old crew was really walk down memory lane and listening to all the hardcore science. I have since moved on from science, and I'm in marketing at Illumina and do sequencing. But I also worked on Chlorobium tepidum, so it was really great to get a little refresher on that. And I worked on the Fenner-Matthews-Olsen protein, and I think Olsen came to the lab one year, and we worked together several times. And I definitely grew bacteria in the cabinets, but we also grew them in the big cylinder that was about as big as I was. And growing it was great, but spinning all those cells down took probably 7 to 10 hours, continuous, running both centrifuges in our lab and in Jim Allen's lab. And my project was to try and establish a site-directed mutagenesis system. And I'm sad that it hasn't happened. Happy for me that, I look back on those years, those were some tough times. And Bob was always very encouraging. And he says, keep going, Diane. He'd always be very positive. Didn't micromanage and kind of let us do and find other things that we could do to support our work. And I heard about the couch. So in our lab, we had an old pastel couch. I believe that used to belong to you guys in the lab, yeah. And every day Bob would come down with a white plastic bag, and his lunch was an old banana and a yogurt. And he would sit down on the couch every day, and it was just a great time. And I always remember Bob in a great mood. I don't remember seeing you in a bad mood, but my husband, Michael, who worked for you for 9 years, he remembers times when you were in a bad mood. And I also have to say, thank you for employing him. He was my then boyfriend, and I said, we've got this, person I think would be fabulous for the lab. And you said, well, okay, but if you two break up, you still have to work together. And we're celebrating our 18th anniversary this year. So thank you so much, Bob. Congratulations on your retirement, and same to you, Liz. And the kids, I remember you guys growing up and going to the house, hiking and swimming in your fabulous home in Ahwatukee. Fabulous memories, and thank you so much, and congratulations.
Hi there, I'm Sam, Bob's son. My father sometimes jokes that whenever anybody asks when he asks anybody to guess what his profession is, they immediately go with professor. We believe it's a combination of the sweater vests and the hair. But that's only on the surface. My dad was born to be a scientist. I've heard plenty of stories about his first chemistry set, mostly about making stink bombs to mess with his older brother. Obviously, I wasn't around when he first cracked open that old chemistry set, but I can perfectly picture the glow on his face, only some of which was caused by the uranium they were giving to children back then. I know what that glow looked like because I've seen it for most of my life. Whenever my dad learns something new and interesting, his face splits into a huge grin and he exclaims, "Cool!" I've seen it when he's reading scientific publications. I've seen it when we're watching River Monsters, despite the protests of my mother. I've seen it when he reads some silly clickbait article, and I've seen it when we watch old family movies. That glow is the delight of discovery, the light of curiosity, and it's what makes him such a talented scientist. For my dad, work was never really work because he was always doing what he loved best, learning new things about interesting topics. He may be retiring from the life of a professional scientist, but that love of learning is never going to leave him, and that deep curiosity about the natural world is the most valuable gift my father's ever given me, and I'm deeply grateful for it. My sister and I are no longer the vibrating children we once were, bouncing off the walls of his lab and begging for more dry ice to make spooky fog. But I'll never forget the science lessons he gave us way back when. My parents are moving back to Arizona to be nearer to us, and I'm very much looking forward to continuing down the path of learning you set me on so long ago. I couldn't ask for a better guide. Love you, Dad.
Come on now. Choose to open it now or wait. have one that is a album of all the photos that everyone sent in. And if folks have signed it, that's good. If you haven't, please wander down and feel free to continue signing. And then I'll back up to biology. Well, I got it. You have to move for this. That's part of the process. So something for us you to remember everybody. Thank you.
All right, I said I would go first, so Bob would have the last word. I thought I would just catch you up a little more on the meaning of that part of the slide that you saw before, how Bob and I actually got together. We both grew up in Nebraska. Some of you know this story. Lived about 30 miles apart, but had never met. But it turns out that my great aunt and his mother were best friends in high school and later taught in the same high school. So one day I was at my grandpa's for Easter, and my great aunt learned I was driving to Berkeley alone to start graduate school. And she said, "Oh, that's too far." She said, "You know, my friend Jane," and I knew who that was because she talked about her all the time. My friend Jane's son, Bobby, he is going to fly to California to Berkeley to grad school in chemistry. So why don't you ask him if he'll ride with you? So I said, I don't know him, but yeah, okay. So I called him on the phone, said, "You want to come down and then ride to California?" He said, "Yeah, okay." So I had met his brother once, but I had never met him. So he drove down. No, he took the bus, right? The bus down to Topeka, Kansas. And I picked him up at the bus station. And I had my stepsister's little dog with me, and he was afraid the dog was coming along, which it was not. So he slept on the couch. We left the next morning for graduate school at Berkeley. And what was it, about less than a year later, we got married. So that's the thing. So that was what you saw. This is our wedding in Golden Gate Park. We walked all over the park and found the place we thought was great. It turned out you weren't supposed to have weddings there, so we just had it anyway. We weren't there long. And so we had a potluck picnic in the park for our wedding. It was a really fun time. So you can see somebody gave us an ice cream freezer. That's what's sticking out of Bob's hand up there. And so it's been a long time. That was 1971, yes. So here we are. The two great kids are here. And so now I'm going to turn it over to whatever you want to do.
Well, this is something that I'm not usually in a position of, which is being at a loss for words. This has been really one of the most remarkable days of my life. You don't normally go someplace where people say nice things about you for an entire day. I know it'll never happen again, and I appreciate so much all of you many of you have come really a long ways, and I just couldn't be more thankful for all the wonderful people that I've had in my group over the years, and it's just really an emotional moment for me to be standing up here. So one thing I do want to do is to remind all of you that tonight there is a party at our house, which I think I sent most of you an email last night. If you don't have it, let me know. It's at 7:30 tonight at our house, which is about a mile from here. And so those of you who have cars at the Night Center can hopefully give rides to some of the others. I don't know how many people have cars, but anyway, we'll try to work that out one way or another. Yeah, you could always Uber. It costs about, I don't know, $3 or $4. Yeah, and so there'll be a lot of food and drink, and hopefully a good time. We can continue all the discussions and so on this evening. And so it's just so amazing to me to see all of you people who have come so far. It's just overwhelming to me. So thank you all again, and I appreciate having had you all in my lives. I always say that my students are like my children. I mean, I have 2 real children, but I have, I don't know, 75 or so, whatever kind of children. So thank you all again. Bye-bye.